Good morning and welcome, everybody. I'm delighted to welcome you to the Amyloidosis Forum meeting on imaging. Thank you so much for joining us today, both in person and virtually. I'm Isabelle Lousada, and I'm the CEO and founder of the Amyloidosis Research Consortium. I myself am a patient. I was diagnosed 25 years ago with AL amyloidosis. Next slide. In 2019, the FDA invited ARC to form a public-private partnership with, and ARC is the Amyloidosis Research Consortium, which we founded, as I mentioned, in 2015. The public-private partnership has been called the Amyloidosis Forum, and the focus is to identify and bridge the scientific gaps that are acting as barriers to drug discovery and the development for treatments of amyloidosis. To date, the forum has focused on AL amyloidosis, and we're delighted that we're now expanding the brief to incorporate ATTR amyloidosis. Next slide. To date, we've actually had 3 meetings. The inaugural meeting, which was held in 2019, was on understanding AL amyloidosis. And one of the priorities that was raised in that meeting was around imaging. So I'm thrilled that we're moving on to imaging today. We've had a series of meetings, formed working groups, and done a lot of work and had a number of publications on novel endpoints and analyses in multi-system rare disease trials and considering endpoints, novel endpoints for AL amyloidosis. The next slide. The planning committee has done an incredible job at putting together a thoughtful, thorough, and thought-provoking agenda for this meeting, and has been chaired by Dr. Sharmila Dorbala, who is professor of radiology at Harvard Medical School, director of Nuclear Cardiology at Brigham and Women's Hospital, and director of the Amyloidosis Research at Brigham and Women's. She has done an extraordinary job, and without her, this meeting would not be nearly as impressive as I think it will be today. My thanks also go to Dr. Adigun, who serves as the FDA Forum liaison, and Dr. Dunnmon, who was the initial liaison, who is joining us today in a different role, and was really instrumental in establishing the forum. And Dr. Rahendran, who really helped shape this meeting, so that the planning committee have really worked hard to shape this meeting. And in particular, great thanks go to ARC's Christophe, who most of you have engaged with and has really shepherded this meeting together. The goals of today's meeting are to gain alignment on what is needed to enable the use of imaging measures as tools and trials for new therapeutics for AL and ATTR. Next slide. With the following: determining study eligibility, assessing safety throughout the study, uses as clinical endpoints for regulatory decision-making. And from this, we intend to identify work streams or working groups to focus on key efforts to standardize and or validate imaging techniques to make them fit for purpose. We have a very full agenda. Next slide. And for those of you in the room, you should have received a copy of it. For those of you who are watching remotely, there should be a link that will be put in the chat to the agenda. We have the next slide. And the morning session will be moderated by Dr. Dorbala and focus on imaging biomarkers. With the next slide. The afternoon session will be moderated by Dr. Dunnmon and focus on pathways to surrogacy and qualification of imaging measures. So just a few housekeeping questions. Questions will be addressed within each moderated discussion. They're pretty tight on time. There'll be a small section for designated Q&A. At the end of the day, there'll be a larger section of time for Q&A. if you're going to ask a question, please turn your mic on and use it, or if you're in the back, please go to the standing mic. Otherwise, people on the virtual meeting won't be able to hear. And if we run out of time, any questions we will follow up with afterwards if they've come through the chat. We have a few planned breaks through the day, and we started on time and we intend to end on time. There is, for anybody who's in here, the Wi-Fi is FDA public and the code is public access. Still not working. Next slide. I want to thank our supporters today, and I'm going to hand over to Dr. Adigun.
Thank you very much, Isabelle. Good morning. My name is Rosalyn Adigun, and I am a medical officer in the Division of Cardiology and Nephrology Drug Products in the Center for Drug Evaluation and Research, as well as the CDER liaison to the Amyloidosis Forum. It's my pleasure to welcome everyone to the White Oak campus, both our in-person and virtual audience for today's meeting. I have no disclosures, and here's my disclaimer. The findings and conclusions in these presentations have not been formally disseminated by the Food and Drug Administration and should not be construed to represent any agency determination or policy. The mention of commercial products, their sources, or their use in connection with material reported herein is not to be construed as either an actual or implied endorsement of such products by the Department of Health and Human Services. The Critical Path Initiative, launched in 2004, is FDA's national strategy for transforming the way FDA-regulated medical products are developed, evaluated, and manufactured. Through collaborations with various stakeholders, these initiatives seek to foster innovation and bring efficiency into the FDA's regulated product development process. One of the mechanisms within which CDER works with stakeholders are through public-private partnerships. These partnerships are established to address specific regulatory science gaps or needs that are beyond the capacity and/or resources of a single organization. CDER and stakeholders leverage individual expertise and resources for the conduct of mutually beneficial scientific activities in the pre-competitive domain to achieve a shared goal. These efforts are also aimed at bridging scientific gaps in drug discovery and development. We aim to promote the development of research tools, platforms, clinical databases, methodologies, and predictive models to advance knowledge of disease and safety profiles of drugs. It is our hope that through these activities from the public-private partnerships that they benefit the public health. Slide. CDER is engaged in a number of public-private partnerships and consortiums, as you can see in this slide. The Amyloidosis Forum specifically, established in 2019, has actively engaged various stakeholders, most of whom are present today, in efforts to facilitate collaboration and innovation in medical product development in the area of amyloidosis. Of note, to guide our discussions today and also to describe the limits of CDER's engagement with PPPs. Next slide, please. CDER is limited to providing a general perspective on regulatory standards, scientific issues, and scientific gaps related to pre-competitive drug development. And as such, CDER staff cannot comment on any specific regulatory applications, products, or other non-public information. We also cannot provide specific opinions on the quality or quantity of scientific evidence for regulatory decision-making. We cannot provide opinions on what conclusions an official regulatory review might reach based on available scientific evidence, or provide recommendations or comments on submissions intended for the FDA review team. We also cannot give advice on a specific proprietary drug development program. But we can do a lot of other things. Go to the next. So it's my pleasure today to introduce Dr. Sharmila Dorbala, who is going to walk us through the morning's agenda. Dr. Dorbala is the Director of Nuclear Cardiology at Brigham and Women's Hospital and professor of radiology at Harvard Medical School. As a cardiovascular imaging specialist, she practices cardiac SPECT, PET, and echocardiography, and her research is focused on molecular imaging in the area of cardiac amyloidosis. Dr. Dorbala is a past president of the American Society of Nuclear Cardiology and of the Cardiovascular Council of the Society of Nuclear Medicine and Molecular Imaging. She's an associate editor of the Journal of Nuclear Cardiology and a past associate editor of Circulation Cardiovascular Imaging. Dr. Dorbala has authored or co-authored more than 250 articles, abstracts, and presentations. She is passionate about teaching nuclear cardiology and has given more than 200 presentations at medical society meetings, regionally, nationally, and internationally. She served as a principal investigator for several clinical research studies in cardiac amyloidosis imaging, and she has received research funding from the National Institutes of Health, the American Heart Association, Foundation, philanthropy, and industry grants. I look forward to hearing your insights on the central role of imaging-based biomarkers as she guides us through this morning's sessions. I will now turn it over to Dr. Dorbala.
Thank you very much, Dr. Adigun. First of all, I would like to also extend a very warm welcome to each of you who's attending this meeting in person and virtually. And my thanks to ARC, Christian Shu, and Isabelle Lousada for bringing together really innovators in amyloidosis from academic centers, from clinical practitioners, and from industry together with regulators to make this meeting possible and to move the field forward. These are my disclosures, and I wanted to just start off the day by giving some background about amyloidosis. As we all know, misfolded proteins accumulate as non-branching beta-pleated sheet structure fibrils in various organs. These amyloid deposits demonstrate congophilia, and they demonstrate apple green birefringence under polarized light. Systemic amyloidosis is a disease that affects multiple organs, either through mass effect or through toxic effects from the protofibrils or oligomeric forms of these misfolded proteins. Although there are several different types of precursor proteins that can cause the disease of amyloidosis, the two main forms that affect the heart are the amyloidosis light chain, which is an immunoglobulin light chain misfolding from a plasma cell dyscrasia. The other common form is called the transthyretin amyloidosis, and transthyretin is a transport protein produced by the liver mostly, and if it breaks down, either because of aging-related insults or because of an autosomal dominant mutation, it can cause transthyretin amyloidosis. This is particularly important because amyloid deposition in the heart, whether it's AL or ATTR, is almost always the cause of death in most of these patients. Amyloidosis, especially AL amyloidosis, is a very rapidly progressive disease. As shown in this slide, data from the Mayo Clinic and several other investigators have shown similar results, showing that even with plasma cell therapy, the median survival of these patients is only 24 months. And on the right side, you see that before the advent of TTR-directed therapies, TTR amyloidosis patients had a median survival of only 3.6 years. If you put it in context, these are somewhat similar to cancer literature, and this is a very serious illness. We now fortunately have new therapies. Daratumumab has been approved for treatment of AL amyloidosis. This has significantly improved survival compared to control patients who received standard of care Cybody therapy at that point. Despite that, you'll notice that at 12 months, patients who received Cybody plus daratumumab had nearly a 20% mortality in 1 year. And patients on placebo, the control group patients who received standard therapy had more than 30% mortality. So this is a very, very serious illness. We need to be doing more to improve outcomes in this disease. What about TTR? We now have approved therapies. Tafamidis has been shown to improve survival and reduce heart failure hospitalization compared to placebo. However, at 33 months of follow-up, nearly 30% of the patients on tafamidis experienced mortality. And there's now emerging data suggesting that TTR silencing therapy also improves outcomes in cardiomyopathy, and that TTR stabilization therapy may work even in patients without heart failure symptoms. But still keep in mind that current mortality rates remain unacceptably high. We're in a good place with multiple therapies, but we can be doing more. So to really understand the relationship between disease and outcomes, I wanted to start off by sharing with something about atherosclerosis, a disease that we're all most familiar with. Atherogenic risk factors combined with lifestyle and genetic risk factors result in atherosclerosis, which leads to poor outcomes. How does this happen? This is mediated through multiple signaling pathways that result in cellular changes, metabolic changes, structural changes, and functional changes in the affected organs. All of these together lead to multi-level changes that influence the ultimate phenotype of this disease and outcomes. Now let's extend this to amyloidosis. This is a disease of protein misfolding with amyloid formation. As we discussed, mass effect and toxicity could lead to heterogeneous clinical manifestations in this disease. But if we extend our experience from atherosclerosis, we could expect that this also triggers signaling pathways and a variety of cellular, metabolic, structural, and functional changes, which eventually determine the final phenotype in this disease and the heterogeneity of this phenotype that we are seeing in these patients. As opposed to atherosclerosis, where we have extensive animal models of disease looking at acute ischemia, chronic ischemia, hibernating myocardium, stunned myocardium, where we could study each of these changes in great detail. We also could use imaging to study effect of treatments of atherosclerosis on changes in phenotype, function, metabolic, and cellular changes in amyloidosis, in atherosclerosis. The challenge with amyloidosis is we don't have animal models. Therefore, our knowledge of the cellular and metabolic changes is highly limited at this point. We'll hear as the next talk about the status of the animal model research at this point. But what we know is we can use imaging at this point. We can use imaging to study in great detail the changes in cardiac structure, changes in cardiac function, and in fact, we could even study in changes in cardiac metabolic processes using some of the positron emission tomography agents and looking at microvascular dysfunction in this condition as well. So what affects the heterogeneous phenotype is the type of amyloid, whether it's light chain or transthyretin, the amount of amyloid mass, whether it's a little amyloid in the heart becomes symptomatic, or a lot of amyloid in the liver may become symptomatic. It's the type of the patient, the age of the patient, sex of the patient, other comorbidities, hypertension and others. The stage at which the patient is diagnosed and delay in diagnosis can all affect the ultimate phenotype of this disease. Fortunately, we now have several potential endpoints that we could be using for these clinical trials. We could directly image the disease pathology or the disease process itself by radionuclide tracers, PYP, DPD, HMDP, or amyloid-binding PET tracers, which we believe currently demonstrate amyloid in the body. We have exquisitely sensitive and specific techniques to study minute changes in myocardial structure and function with echocardiography, magnetic resonance imaging, as well as positron emission tomography. And of course, survival, quality of life, 6-minute walk test, and cardiopulmonary exercise testing are useful metrics to evaluate outcomes. The challenge is with ongoing studies, survival as an endpoint is becoming very, very tough to attain. However, this is a deadly disease, and if you wait for survival or if you wait for mortality as an endpoint, we may have lost valuable time where we could be treating these patients with different regimens that could potentially improve patient survival in this case. So several novel therapies are now available, so we're very fortunate. This is not a complete list. I put out few of the treatments that are approved or investigational on this drug, only to point out that your study endpoint may relate somewhat to what you're treating. If you're treating the precursor protein, perhaps you could look at serum free light chain levels. You could look at serum biomarkers. And if you're looking at treatment of ATTR amyloidosis, perhaps we could look at changes in PYP, DPD, echocardiography, or magnetic resonance imaging. If you're looking at fibril-degrading therapies, we could directly image the fibril, possibly with the PET radio traces. Or you could image the effects of changes in fibril on cardiac structure, function, and other processes. So we have a variety of methods, non-invasive methods, to provide deep insights into changes with therapy. So I just want to end with this slide on how the world of oncology is moving ahead very rapidly. There's so much for us to learn in amyloidosis from oncology treatments. So here's one example of many that I picked, which is early stage or advanced stage Hodgkin's lymphoma, where imaging is performed and staging based on imaging is used to change management at every step. So you diagnose this disease, you give 2 cycles of chemotherapy, stage with FDG PET-CT. Then based on the stage, you either continue 2 or 1 cycles of ABVD, or you add radiation therapy. So this type of dynamic adaptive therapy, I think, is what we are almost ready to implement in the field of amyloidosis. Think of it, it's a systemic disease. This is not affecting one organ. A systemic disease with multiple treatment options and multiple imaging options. So thank you very much. So now we would like to move on to the next part of the session. This will be an animal model for AL and ATTR amyloidosis, and Dr. Christophe Sirac will be presenting virtually.
Okay, good morning, everyone. Do you hear me and do you see my slides? Yes? Is it okay? You hear me?
We can hear you fine.
Okay, okay, thank you. Okay, so thank you very much. Thank you for the invitation and for the organization of this excellent meeting. I will try to briefly explain our AL amyloidosis model in the mouse and why it was so difficult to obtain and why it's still a bit difficult to use it. So, well, you all know the amyloidogenesis process with these 2 phases, the lag phase, during which pathogenic protein will slowly accumulate to form the first amyloid seeds, and then the elongation phase. When enough seeds are formed, the elongation phase will start, and which is more rapid, and to form the mature fibrils and eventually organ dysfunction. So to create a mouse model, we had 3 issues. In fact, the first one was to use a highly amyloidogenic immunoglobulin light chain from a patient. The second one was to obtain a quantity of free light chain which was compatible with the disease, so an excess of free light chain, which is not physiologically normal. And the third one is of obviously this lag phase, the duration of this lag phase that we cannot control, and we even don't know in patients how long last this lag phase, and certainly probably several years. So I will not detail the model we created, but just to tell you that we spent several years, more than 10 years, to finally obtain a mouse model expressing an amyloidogenic free light chain at level exceeding those observed in patients, and these mice never developed spontaneous amyloidosis. So we finally used a seeding strategy. So the seeding strategy is to bypass the lag phase by injecting in the mice already formed fibrils that we we formed in vitro with only the variable domain of the light chain, which is the amyloidogenic part of the light chain. And then here it works. So we have some deposits in some organs that you can see here with Congo red, anti-human lambda, and electron microscopy. So of course, what interests you here is which organs. So we have deposits mainly in heart, but also in vessels and spleen. And to a lesser extent, in some mice, we have also deposits in kidney or other tissues. On a practical point of view, we have two protocols to induce amyloidosis in these mice. The first one is what we call the FAST protocol. So we inject fibrils and then we can observe amyloidosis in few days in the mice, but we do not have 100% of positive mice. So it's a bit complicated to use. And the physiological protocol in which we inject only the variable domain of the light chain, so not fibrils, just a part of the light chain, and then they progressively develop amyloidosis with 100% of positive mice after 6 months. Well, this model is a bit complicated to use because you need the mouse model, but you also need all the protocols to produce the light chain, the recombinant light chain, to produce the fibrils or to inject the variable domain in the mice. However, it was already successfully used in some biodistribution studies. I show you here some amyloid binding molecules that we use in our mouse model. So this is ATO2 and ATO3 from Atralis. And we are currently working with John Wall to see if these mice could be used for in vivo imaging with radio-labeled molecules like ATO1. And the main limit, I should say, of this model is that so far we never observed any organ dysfunction, in fact, in these mice. So we have the deposits, but not the symptoms of the disease. And thank you for your attention.
Thank you, Dr. Sirac. Dr. Alexander.
Yeah, great. So I'm just going to spend a couple of minutes for some updates on ATTR amyloidosis models, and then I think we're supposed to have a couple of minutes for Q&A, so look forward to that. Next slide, please. So I think there's a similar theme in terms of developing in vivo ATTR models. Certainly, there's been a number of groups that have used expression of variant transthyretin proteins in mouse models. And I think uniformly, folks have found similar that there's a lack of amyloid formation with just simple expression. And I think that reflects some of the points that Dr. Dorbala made in her talk in terms of what other factors are needed to lead to that initial amyloid formation. Certainly, I think there's a time component. This is a disease in humans that takes place over many years, most likely, and so we're trying to condense that time of amyloidogenesis into a relatively short period of time. And also, what are the other factors that are related to amyloid formation? So age-related, are there age-related factors, impaired proteostasis, and other mechanisms that are poorly understood, probably contribute to that initial amyloidosis event? Next slide. So more recently, there have been successful models that can show amyloid deposition, cardiac amyloid deposition. And these models really are based on expressing a variant transthyretin protein and using seeding of human amyloid fibrils to cause that initial event of amyloid propagation. So I think the main caveats with the models that are currently available is it's really only a partial recapitulation of human disease. Pathologically, there are evidence of amyloid fibrils, but like Dr. Sirac was saying, for AL amyloidosis, there's not necessarily the physiologic manifestations in terms of evidence of restrictive cardiomyopathy, arrhythmia, so forth, which is really important, I think, for a model if we want to understand mechanism, but also have a model where we can do therapeutic interventions to help with drug development and biomarker discovery. So I think that there's been progress, but there's a lot of work to do in order to continue to mimic aspects of the human disease that can help us with a platform for developing therapeutics.
Perfect. Many thanks, Dr. Christophe, Sirac, and Dr. Alexander. So we'll hold off on the questions until the standardization section. So with that, I would like to introduce my co-moderator, Dr. Preston Dunnmon, and I'm truly honored to have worked with him in developing this program. And he will be speaking next, and he will also be moderating the entire afternoon session. Thank you, Dr. Dunnmon.
Thank you, Dr. Dorbala. I'd like to begin by thanking the organizers, as well as specifically Isabelle Lousada, the CEO of the Amyloidosis Research Consortium, and Dr. Adigun, who is now the CEDAR liaison to this public-private partnership in the forum that's its manifestation. A lot of people over the years from these organizations have worked very hard to bring leadership globally in academia, in industry, and in the regulatory bodies to bring their thoughts to bear on how we can move medical development forward in these diseases that are not easy to do, both for lack of animal models, for necessitating human data, but then then you overlay on that what you have to show the regulators so they can meet their obligations under their regional laws. This gets to be very complicated and a multifaceted set of rare diseases. The thing I'd like to point out here is there's a perfect storm brewing here, because on top of the lack of a validated animal model, what you've got going on here is some drugs now being approved. There's one for AL. There's a drug approved for ATTR amyloid in the heart, and there's several for the neurology indication. So what you've got is this background of additional meds going on that's dropping outcome event rates. This is a common phenomenon across multiple disease classes. Now, when you've got this going on in left heart failure, which we do, there's a lot of drugs out there, it's reducing event rates, but you've still got this ocean of left heart failure out there. It's workable with some difficulty. But when you've got this perfect storm of things brewing around a set of rare diseases, you're looking at the possibility of innovation for these patients essentially stopping. And so I think it's on us, the people in this room, to help try to think our way out of this as far as defining what the future is going to look like. And that's what's really brought this interest in imaging to the forefront, because particularly with the multiple kinds of imaging available and the way these modalities are rapidly developing, there's potentially just a whole lot to be done with this. Next slide, please. Oh, I've got it. All right. So there we go. As far as imaging as a biomarker, okay, because what we're talking about today are biomarkers. They're potentially using non-clinical data to support medical development programs. And if it's not a clinical outcome, it's a biomarker. And so in the confines of both this room and the virtual room that we have here with us, which actually dwarfs our in-person attendance here, Yeah, I was thinking about, this rhetorical question of raising your virtual hand if you've ever wanted to ask, or been asked the following questions. Can an image be a biomarker? Are there different classifications of biomarkers? Can an image biomarker be an endpoint in a phase 1 or a phase 2 trial? And then the really provocative question, can an image be either itself a primary efficacy endpoint or part of a collage of components that would form a composite endpoint for a phase 3 trial? I'm stuck here. Can somebody go to the next slide? Maybe you got the magic finger here. It was late. Okay. No, back up to. Yeah, they're good. So a lot of people think about this, and this is, and so I want to ground the conversation we have today in what the regulators, at least in the U.S., and I'd love to hear from our MHRA and EMA colleagues about this as the day goes forward, about how we're thinking about classifying biomarkers. Because before you can use something and use a tool, you got to figure out what to call it and how it works. And we got to get a common language here of how we're going to do this. And so this reference here is a living document that's a cooperation between the FDA and the NIH that actually does exactly that. This is a glossary, and it's available for your download. The last time this was updated was November of 2021, and it actually defines what a diagnostic biomarker is, something like glomerular filtration rate that you can use to diagnose renal failure. Monitoring biomarkers like the PSA for prostate, disorders, and used as a repeated measure. We'll skip that one in the middle. Let's go down to prognostic biomarkers, where does this biomarker at baseline, forget drug effect, does the biomarker predict outcomes the worse it gets? And depending on which direction your biomarker goes that's bad, say in the heart failure example, NT-proBNP, does the higher that thing go, does that correlate with outcomes getting worse? And the heart failure community has repeatedly shown that relationship. And then there are then predictive biomarkers. Now, you notice here, you'll be forgiven if you get confused on this, because I point out here that BRCA gene mutations are both prognostic and predictive biomarkers. And to make it even more confusing here, this one in the middle, response biomarker, that's talking about response to a therapeutic. And so a drug-induced movement of a biomarker. Well, internally, when FDA talks about that, at least in some of the divisions, they're using the term predictive for that instead of response biomarker, because the drug effect is predicting the outcome. So you've got two lexicons going on in parallel. Next slide, please. And then as far as then once you figure out what you're going to call these biomarkers, how you can use them in a regulatory framework to make them believable as a surrogate clinical endpoint. This slide is verbatim from a presentation in May of this year by Dr. Lisa McShane. You'll notice where she's from at NIH. She was presenting this slide at the Pediatric Oncology Drugs Advisory Committee, and she was addressing this issue of the Prentice criteria, which is typically the go-to way that that people address this issue of qualifying a biomarker. And there are essentially four criteria. You got to show that the biomarker induces a statistically significant movement in a clinical outcome of interest. Feels better, functions better, lives longer. You've then got to show that the drug induced a statistically significant movement in the biomarker of interest. You then have to show that the downward movement in the clinical outcome is attributable to the downward movement in the biomarker, okay? And then obviously at baseline, it has to be prognostic, and the higher the biomarker is, the worse you do. Well, you'll notice that red comment down there at the bottom of this slide where Dr. McShane stated that this is conceptually appealing, but generally impractical because it is so stringent that it is almost never satisfied. I personally am aware of 2 examples in medical history where this has been done successfully. So with that, let's go ahead and proceed on to the next slide. I'd like to thank everyone. We'll talk more about these, the regulatory framework of all of this afternoon, but I want you all to have this in your head as Dr. Dorbala now takes you through the morning session on the current state-of-the-art of imaging.
Thank you very much, Dr. Dunnmon. I think you've perfectly laid the ground for the next session that's to start. So what we're going to do over the next few sessions is talk about the various imaging modalities, starting with echocardiography, MRI, then SPECT and PET. We'll also talk about functional and health-related measures and have a moderated discussion. So what I'll do is, in the interest of time, we'll proceed with the talks and save the questions for discussion, which is embedded into every section. So now I'm really pleased and honored to invite Dr. Rodney Falk to speak about echocardiography in AL and ATTR amyloidosis.
Thank you, Sharmila, and thank you for inviting me here. Excuse the slight informality here, but I took off my jacket because I think it's a hot topic, and my role is to introduce something that will be a heated discussion. Let me just first talk about AL amyloidosis. This is a slightly different talk because I've got other speakers here, and I want to just tease some ideas out. In AL amyloidosis, cardiac involvement is the driving force for mortality, and it's mediated by rapidly progressive heart failure or less commonly by sudden death. So the heart is central here. We know, particularly work from the National Amyloidosis Center with Ashu here, that normalization of affected free light chains is associated with reduced mortality and improved heart failure compared to non-responders. So the pathology of the disease, improving the pathology of the disease, improves heart failure. We also know that at least in severe abnormalities on the echocardiogram, such as reduced ejection fraction, severe diastolic dysfunction, if you see those right at the beginning, it portends a poor prognosis. But they're actually quite a crude measure of outcome, because those, when you look at them, it's really only in the severest disease, and they're not going to be terribly helpful if we see changes with therapy. On the other hand, echo imaging using strain, which I'll talk about very briefly what that is for those who are not familiar, it's reproducible. There's extensive support in the literature for its prognostic value in various cardiac diseases. So if you look at standard echo parameters, ultrasound of the heart, wall thickness, amyloid thickens the walls. It's not very reproducible reader to reader. You would think it is, but it isn't. Stroke volume, cardiac output, you can calculate those, but they're load dependent and they're derived. You have to make a number of assumptions and multiplications and so on, and obviously your errors are going to be increased. Diastolic dysfunction, which is a critical aspect in amyloidosis, as the heart gets stiffer, it's nonlinear. There are various stages of it, but you're either in this stage or that stage or the other stage, but in between there's no real measurements. It may be difficult to assess, for example, in atrial fibrillation. An ejection fraction, the percentage blood that the heart puts out, is load dependent. If the blood pressure is higher, it drops off. and it's actually a crude measure of function. On the other hand, strain imaging, which is the way the heart moves longitudinally, it's independent of angle when you're taking the images. You can directly determine it. You don't have to do any other calculations. The machines that do it now, the echo machines, are semi-automatic or fully automatic and less operator dependent. It's remarkably reducible, and you can look at not just the left ventricle, but the right ventricle and the atria. Let me show you an example of what strain looks like in amyloidosis. On the left is a normal bullseye pattern. It's color-coded. And in amyloid, there's regional variation of strain, and each of these numbers represents a segment of the heart. They're negative, so -3 at the edge there means the heart in that segment moves just by 3% as it contracts, which is not very much compared on the left to the same segment where it's a 22% shortening. Sorry, I missed this slide. Strain imaging is clinically used in multiple cardiac diseases, clinically used. So it's used for the assessment of subclinical left ventricular dysfunction in cardiomyopathy. You have a family member with a familial cardiomyopathy, the echo looks normal, you do the strain, there's impaired movement. That patient is in trouble, and they're the ones who are going to be gene positive. In heart failure with preserved ejection fraction, when all the other factors look normal, impaired strain is going to tell you that something is more likely to happen than in the patient who doesn't have impaired strain. The serial assessment of left ventricular dysfunction, this is really critical in patients undergoing potentially toxic chemotherapy for example, breast cancer. You can have an echocardiogram that superficially looks normal, but is differed from the previous one a month or two before with a couple of months of chemo by the fact that the strain is beginning to deteriorate. That patient is at risk of developing heart failure if you continue, and there are now data that suggest that if you intervene early, you're going to prevent heart failure, and the use of strain is incorporated in the treatment of breast cancer in many national societies' recommendations for breast cancer treatment. It's used as an aid in determining optimal timing in patients who have severe aortic valve disease, in whom the echo looks okay, they don't have symptoms, the ejection fraction is fine, but longitudinally the heart is having trouble. Those patients are going to run into problems. And it is valuable in the diagnosis and assessment of cardiac amyloid, as I showed you there. Just briefly, two slides, and then we'll open for discussion. In TTR amyloid, this is a paper that our group published earlier this year in a group of patients, small group of patients treated with tafamidis compared with historic controls. And after a year, the strain imaging was virtually unchanged in the tafamidis group and had deteriorated in the controls. Look at the numbers, only 23 in one group, 22 in another, and yet you can see a difference in these groups already utilizing this technique. The bottom bullseye is a sophisticated addition to strain imaging which takes into account blood pressure and maybe an even better and easily obtained tool. And finally, here's an example of how strain improves. So in TTR amyloid, you can stabilize it. In AL, you get rid of the light chains. You get improved ventricular function with improved strain. On the left is the strain imaging of a patient before chemotherapy. On the right is 18 months after chemotherapy. The more red, the more pink there is, and this is not a political statement at all, the better you are. And this is quite a dramatic improvement, and it's not an isolated improvement, because if you look at data from the London group, a paper with Ashu as the senior author, what you can see in the top left graph is that depending on strain at diagnosis, the outcome varies over time. If you look at the second graph on the bottom left, the change in strain at 1 year also predicts what's going to happen. So it actually gets better. And on the right, there are proposed criteria for staging which don't just take into account the biomarkers that have been used before, but now look at the addition of strain, both in terms of what happens at baseline and also the change in strain that happens with follow-up. So I think this is a well-validated tool. It's already used clinically in multiple diseases. It's been shown over and over again in amyloidosis to predict prognosis and the changes to predict long-term outcome and to be associated with improvement clinically, and meets many of the criteria that we have heard of. And I feel that this is, and it's easily reproducible and easily done. So I'd like to open to my colleagues, and Ashu is going to moderate this. The question is not just should we be using strain, but are there other markers, and should this be incorporated and accepted as an appropriate tool? Thank you.
Thank you, Rodney, for that wonderful introduction to echocardiography as a tool for both diagnosis, monitoring, and prognosis for amyloidosis. I guess we've been using echo for a very long time in amyloidosis, and we keep talking about the different echo parameters. So maybe what I would do is I'll start with a couple of questions, and then it's open to everyone around the table to start commenting. So maybe if I start with you, Sarah, as one of the panelists on the here. I mean, what is lacking? Why haven't we actually, we keep changing, but it's never really become the major prognostic marker. Our staging systems for biomarker-based. What are the gaps that are in echo? How can we look at it further?
Yeah, I think the gap is just kind of physician commitment and time and the platforms that we use. So training with Dr. Falk, particularly, and obviously with Dr. Dorbala as well, but with Rodney, we sit down and look at the echoes and we do the strain ourselves. And we use the GE platform, which is very user-friendly. So we kind of really built that into our workflow clinically, trained the techs up on this platform. So we're getting it real time in the clinic. We have a trained technologist who's very, very good at interscan variability. So I can get that real time when I'm seeing the patient, and I trust those measurements. The problem is if you don't have that workflow set up, do you go yourself, go back, measure the strain yourself? Have you done it on the correct ultrasound machine? Do you need to go and use another vendor that mightn't have the same reliability and interscan variability? So I think that's really where the gap is that kind of vendor-neutral platform that can do it quickly and reliably, that we can really use it real-time clinically. And we've kind of got that in our workflow, but I think other centres, it's still a major obstacle.
If I could just add to that, I'd say we have a very similar process at our centre as well, and I think within our own centre, I think we feel pretty confident trusting measurements along the time point for each individual patient. But I think one of the issues that comes up is if patients go between centres, so if they're followed locally and they come back to your centre, or they get their treatment elsewhere and come to your centre, then it's more difficult to know whether the variability is going to be the same from one centre to another. And then there are these issues with different vendors, which hopefully I think soon will become a little bit better, a little bit more vendor neutral. And there are ways to address that, but it's time consuming because it means then you need to take the images from one vendor, put it into another kind of vendor neutral platform in order to do the analysis. So that I think that's probably the major gap in being able to use it more widely.
And I guess I'll go to Cristina next just for a question in terms of, you've been involved with some of the trials that are being done for amyloidosis. How difficult or easy have you found to kind of incorporate this tool across you're using it globally across sites around the world?
So for echocardiography, I think that it's still a very widely available technique. So it's really a matter, as been said, to implement the correct standardization for a clinical trial, and of course beyond the setting of the clinical trial, then in the clinical practice. But for strain, for instance, what we have found very useful has been the use of a centralized laboratory that reads all the echocardiograms, that performs all the standardization, blinded to time. So it's really something that I still believe for echocardiography is very simple to, or potentially simple to be implemented. When we go beyond echocardiography, then of course the things change, and we will discuss later. But I think that from a gap perspective, it's not just about the technique, but it's also about what all these parameters, measures really reflect when we look at a disease. So, for instance, I think there are still a lot of debates about what really strain does reflect in cardiac amyloidosis, right? So we see that is a very good prognostic marker, that changes are correlated with outcome, but then the question why we cannot really implement this yet as a surrogate endpoint is, I think, based on what is reflecting when a patient is undergoing a certain treatment. Does it change if the patient is undergoing a standard anti-plasma cell dyscrasia therapy or an anti-fibril amyloid remover? So these are still questions that are not fully addressed, and I'm glad that all these newer clinical trials are implementing all these different techniques to answer these questions.
That's a very important point. And I guess there's one point which I would kind of open up to the panel. I mean, I'm not a cardiologist, so I put this disclaimer. I'm a hematologist. So if my question sounds silly, I'm the user of your results rather than actually somebody who does it. There are changes in strain, and when we did our analysis of strain in our AL population, we were quite surprised to see that the changes that we saw weren't necessarily the same across the different groups. So if your strain was better, the change wasn't the same as if the strain was much worse when they started. So it's a question to everybody. When we're using strain, both for prognosis and for monitoring, is there a linearity in there? Is that something that we need to understand and take into account based on how severe the heart is affected? So, for example, a TTR heart with a 20-millimeter thick wall, will the strain behave in the same way as an AL heart with a 14-millimeter thick wall? It's open for anyone to comment. Maybe Rodney, you can start, and then I'm going to get over to Matt after.
Well, I think we don't. AL and a TTR are different diseases, and and it's 30 years since we showed basically that patients do much worse with AL than TTR, even though the echo looks the same. So I don't think you should talk about as though it's one disease. But I actually going to do what a politician does and change the topic slightly, if you don't mind. I just want to make the point that we have to think differently between using this in clinical trials, is it a good endpoint, and using it clinically for our patients to see how they're doing. I think that in clinical trials, as Cristina points out, if you have a good central lab and a reasonable quality echo from your sites, that this is an extremely important tool that I believe can be a surrogate. That's, I think, totally different from using it in the clinic. We don't know exactly what that will mean if somebody has improved, except, yes, you've improved, and they're probably going to do better. But in order to use it as a surrogate endpoint, which, as you've shown, Ashu, really predicts what's going to happen in a year or so to the patient, are they going to survive or not? Are they going to be hospitalized or not? That's really important, and that I understand is what this panel is about. It's another leap of faith to use it clinically, but I don't think we need to do that yet. The real question we have to address is, should it be something and can it be something that's introduced into clinical trials? And I agree with Cristina that it's easy to do, and with a good central lab, it's a very useful tool.
Thank you, Rodney. That's a very important point. Now, I see Marianna's hand up there. So, Marianna?
Yeah, I just wanted to make a comment because I completely agree with Rodney that thinking in the sense, one thing is thinking about the imaging modality, being able to track changes at the individual level. And one thing is actually for the imaging modality, a specific parameters to be used in clinical trials, a very, very different concept. And so when we think about using, for example, strain into clinical trial, the massive advantage of echo is that it's done absolutely everywhere, both in clinical practice and research. And I think that this is a massive advantage of echo compared to any other imaging modality we are going to discuss today, and is absolutely undeniable. So what we really need to do is we really to get down on a better precision and lower the variability in two phases, image acquisition and interpretation. And I think one of the things that we need to keep in mind is that from image acquisition, of course, expertise is going to be crucial, and we're never going to get out of the importance of training expertise in image acquisition. But in terms of image analysis, I think in the next 3 years, we will completely shift from the from the very well-known model of core lab analysis to actually implementing AI in clinical trials, in the sense what's happening now, I think in echo especially, much more than in other imaging modality, I think is a complete revolution from this point of view. And I'm absolutely convinced that maybe in 3 years, the standard to measure certain parameters is not going to actually be anymore as going there and taking the whole thickness of everything, but actually AI will be able to overcome many of these limitations, and by increasing precision and lowering variability will significantly increase the signal-to-noise ratio. And this could take a modality which is widely available everywhere really to the next level, whereby almost is going to be one of the most important to me, because you managed to get down on really variability, improve precision, and improve the signal-to-noise ratio. And so I think in the next 3 years, there will be massive steps forward in this respect.
Thank you, Marianna. That's a very important point, and I was about to come to that next. I think Sarah wants to make a comment.
Yeah, I was just going to echo exactly what Marianna said. There is already AI out there that was shown in the WAVE study published by Roberto Lang, looking at COVID patients, where they're using that AI technology in strain was able to detect kind of a much more meaningful change rather than human interpretation. So I think it's probably there. It's just the case of getting those platforms implemented into our clinical work, and using those platforms. They're already FDA cleared for clinical trials.
Marianna, I agree with you. I would point out, though, that sort of maybe it's not quite AI, but when you're measuring strain, unlike virtually anything else in echo, it's a click, 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 and the computer internally makes the measurement. I'm not taking my calipers like I'm doing for the wall, or tracing something to look for the ejection fraction. So that's why it's so robust and reproducible. So that's another positive for that. I just, if I could ask you, throw to the panel, may I take your, just as a quick question, is there anything else that we should be thinking about in the echo that may be reproducible?
So I was just going to go to Matt and Sharmila quickly, and then we'll come back to your point.
I would just ask you, Ashu, as the only person yet at this table who's published widespread data from a large center that shows that it has prognostic value, and we hear about core labs. Do we need a virtual core lab for the community? I mean, with AI that we're talking about, and providers having a great deal of difficulty getting these measurements done, is there some, international mechanism to, upload these studies from various centers and, determine whether or not there's really more widespread prognostic validity in the community rather than just at a single or several expert centers?
I think that's probably a key point, is to validate the results that we have published on a more global scale. And I think that's something that we should try and plan and do, and we would figure out a way that we can do, because there'll be multiple platforms, different ways the studies are done, and they'll need to be analyzed. But validating these results and using those in routine clinical practice so that they can then be used as endpoints, surrogate endpoints, is quite crucial. But yes, absolutely, we do need some validation from what we've seen in a sort of fairly large cohort, though it's still a single center cohort. Sharmila.
Actually, I see Dr. Omar Abu Azzedine raise his hand, so I'll let him go first and then make a comment.
Omar?
Thank you, Dr. Dorbala. Sorry, just very briefly, I fully agree with Marianna and Sarah on this point of AI and the future of echo. I just wanted to engage Dr. Lin and her insights, because she was very involved in the echo core lab as far as the heart failure network trials were concerned. And I just wanted to sort of tap into that a bit and hear more about what challenges did she face, or did the Echo Lab, the core Echo Lab face in those trials. I think that would be insightful.
Thanks, Omar. So I think that comes back to Rodney's point then about how you use this clinically versus how you use this for research. So the issue of variability, I think you can control a lot better as it pertains to strain if you're talking about kind of core lab interpretation of large amounts of data from multiple centers. Because, you know, one thing you can do is you can eliminate the concern about vendor differences by putting everything into one specific type of vendor that can be used for analysis. The other thing is you can also limit the number of people that do the interpretation, so you can really cut down on the variability. So if you're looking at large populations and research and clinical trials across multiple centers, I think that's easier to control. I think the clinical question is you know, how you use it clinically and how you control for the variability clinically is probably the bigger gap to be addressed, and then perhaps not so relevant to this discussion today.
Go ahead. Thinking about biomarkers, you know, is it important to differentiate biomarkers that reflect or measure the effect of disease as opposed to those that directly image the biology of disease, right? So strain is measuring the effect of the disease on LV function. So how do you interpret that in somebody who has aortic stenosis and amyloid, somebody who has amyloid and atrial fibrillation, right? So changes may be due to changes in the disease we're interested in or in one of the comorbidities, as opposed to using something like a PET tracer that actually measures amyloid. Something to think about.
I think that's a very key point, because these are often older patients with multiple comorbidities. And I mean, this may not necessarily be directly relevant to clinical trials, but in amyloidosis patients, we often need more than one measure to tell us what's going on. And I think it becomes a bit more tricky in clinical trials when you're putting in multiple measures, but it may be a multiple modality endpoint rather than a single modality endpoint that we may need to be working towards. Because we have got MRI, we've got PET, we have got echo, and we need to use all of our available tools to assess what's going on. I think echo has an advantage. It's easily available. It's done everywhere. It's widely reproducible. You can do it as often as you like, whereas with some of the other tracers, there are sort of challenges how often you can repeat these imaging. But your point is very valid. Echo is looking at the function, which is an effect of what amyloid has done to the heart rather than the actual amount of amyloid in the heart itself. So it's a surrogate for the amyloid damage to the myocardium. With MRI, maybe you can actually see the expansion of the interstitium, which is telling you where the amyloid is and how it is changing. But again, MRI tracers are not directly binding to the fibrils like PET tracers are. So PET is looking at something quite different to what the MRI is looking at, which is quite different to what the echo is looking at. So when you put all three together, you're looking at very different things. I mean, we've talked about echo quite a bit, but one question which Rodney raised, which I think I'll bring back quickly because I think I can see we've got 3.5 minutes left, is we've talked about strain. Is there anything else in echo that we should be looking at, or should we be focusing only on strain as our key endpoint? Because at the end of this day, we need some sort of take-home message of what are our next steps, how are we going to use this forum to take the next steps towards building on biomarkers with, say, echocardiogram.
I think it's probably Matt's point to try and kind of leverage one of these AI companies potentially to integrate all our imaging, though there's a lot of barriers with sharing imaging and stuff like that, to see if we can replicate your data for that long, like the kind of prognostic and longitudinal change that we see in these populations.
And I think Rodney made a great point about myocardial work indices. I think those would be something that could be hopefully integrated into clinical trials as well.
Sorry, I understood.
The myocardial work indices that you touched upon.
Yeah, so myocardial work, it's just really a more sophisticated measure of strain. You incorporate the blood pressure. But Prem, your point is well taken, except that if you're thinking clinical trials, you're going to exclude patients with aortic stenosis. Yeah. You can exclude patients if you wish, more difficult with atrial fibrillation. And we can do that. That's what we have to do in clinical trials. So this is a tool that is looking, as Ashu says, at function. And at the end of the day, what kills you is worsening function of the heart in this disease. And what stabilizes you is stabilized function or improved function. And everything else, even if you use imaging such as MRI, as I mentioned before, MRI extracellular volume in TTR is not the same as in AL. For the equivalent amount, you may be near dead with AL and functioning well in TTR. But here it's function.
Yeah, I just had one comment. When you're talking about treatment trials and longitudinal imaging, sorry, Rick, I didn't.
Totally fine.
I think as patients get sicker, you'll see that they can't go through SPECT or PET or MRI, and echocardiography is always there. Yeah. So I think I look at this modality as very valuable in providing information about the sickest patients. Rick?
Completely agree with that point. I wanted to ask Ashu and Rodney, in your experience and everybody else, the number of images that are unevaluable, because that's another important consideration. I mean, in my clinical experience with echo, there are a number of times where the image quality precludes really accurate strain assessment, and if a patient is going to be included in the trial and it's going to be an endpoint, the endpoint needs to be accessible. Can you comment on that?
So I'll make a quick comment, which is an uneducated hematology comment, and then I'll give it to Rodney for the proper one. We surprisingly find that that's often a problem where the heart is not that involved with amyloid. When there is amyloid in the heart, the images often tend to be much better. So it's quite interesting that in the non-cardiac patients where the heart isn't that damaged, we often struggle with the good quality images, whereas in amyloid, where you have a thick wall, bright heart, the images are not that bothersome. But Rodney, you can comment on that.
Absolutely agree. This is a disease which is designed, as it were, for strain. They're thick, they're echogenic. Atrial fibrillation is a problem because of the beat-to-beat variation. But otherwise, clinically, we can get answers in 90%, Sarah, would you agree in our lab?
Thank you so much. Really excellent discussion. I'm sorry we will have to move on. We can ask questions during the next session, standardization. We have an entire half an hour for questions. So thank you all. Really amazing discussion by all the panelists. Thank you very much. So the next talk is going to be about cardiac MRI, AL and ATTR amyloidosis. This will be done virtually by Dr. Marianna Fontana. Marianna?
1 second. Yeah. So thank you very much for the invitation to speak. I am presenting the slide on behalf of a group of experts. We all contributed to put together these slides. So me, Frederick, Ruberg, Justin Grodin, Michael Jerosch-Herold, and Michela Brambatti. So MRI uses magnets and magnetization to create an image of the body without ionizing radiations. And we can therefore accurately assess myocardial mass, volumes, whole heart wall thickness, systolic function, myocardial deformation. We can assess also flows and myocardial perfusion. And so CMR on these measurements afford similar information to echo, but with higher reproducibility because of better image contrast, edge detection, and capacity to image tomographically. So basically, we're not constrained to imaging planes limited by the skeletal structure. However, just a second, maybe. However, the key advantage of CMR in cardiac amyloidosis is its unique ability to provide information about the tissue composition by what we call myocardial tissue characterization. And this is because healthy and pathological myocardium have different magnetic properties without contrast, so T1, T2, and T2 star, and with contrast, so LG and ECV. And what we do with legal enhancement, which is the basic technique that it's the cornerstone of all diagnostic pathways in CMR, is that we give gadolinium-based contrast agent. And by the pattern of distribution of the gadolinium within the myocardium, we can assign a specific pathology in a process known as phenotyping. And you can see that the pattern distribution is very different in different myocardial pathologies. But what we are really doing in this process is trying to understand what the core myocardial processes involved are, and especially those processes that are tightly linked to treatment, to the disease, and that can be changed with therapy. And in this way, by focusing on these processes, fewer observation and the first subject are necessary to identify changes, as we are imaging the specific pathway which is tightly linked to the treatment. And the very important role from a diagnostic point of view of the technique of leg enhancement in cardiac amyloidosis has been recognized, and that's why this has been inserted in the consensus documents. This is just one of them, published last year in the European Journal, where the importance of leg enhancement by CMR has been recognized. But so when we see a patient for the first time, what we want to know in this specific setting, does the patient have cardiac amyloidosis or not? And this is an accuracy question. But when we see a patient for the second and third time, the question is completely different because it has nothing to do with accuracy, but has got to do with precision. So if you look at these two pictures, these are two leg enhancement pictures taken from the same patients, but 3 years apart. These are patients with ATTR amyloidosis V122I. So from these pictures, anybody doing MR can say, yes, definitely, this patient has got cardiac amyloidosis. But no one, just based on these pictures, can tell if the patient is better or worse. So leg enhancement is very accurate, but is not good to track changes over time, so for precision. So what can we do to do that? So T1 mapping is a CMR technique that definitely can help into this. This one is a non-conscious T1 map, so just for one breath hold, and then we get one of these maps, and it's a pixel by pixel quantification of the T1, which is an intrinsic myocardial property, but is a composite signal from the myocyte and extracellular space. And because the amyloid sits in the extracellular space, what we want to do is to isolate just the signal from there. And we can do that by giving contrast, which goes just to the extracellular space, repeating the same map again one breath hold after contrast, and we get one of these, which is an extracellular volume map, so pixel by pixel quantification of the extracellular volume. And why is the extracellular volume important in amyloid? Because the amyloid sits in the space between the cells, so we'll expand the extracellular volume. So the extracellular volume is really, in these patients, a measure of the amyloid burden. And this one is an example of why it's important in clinic. These are two patients with ATTR cardiomyopathy, same increase in wall thickness, same degree of systolic and diastolic failure, so we might think they must have the same amyloid burden. But if you look at extracellular volume on the right, the patient at the top, the extracellular volume is 39%, so mild to moderate infiltration, whilst the patient at the bottom, is 69%, 62%, so severe amyloid infiltration. So same structure and function, but very different amyloid load. We can do the same also in the liver and the spleen, and this has been validated against the SAP scintigraphy, which is the current gold standard for visceral deposits. So with one technique, we can measure structure, function, and amyloid load in the liver, the spleen, and the heart. And because ECV is measured the amyloid, is not surprising that does correlate with prognosis in both AL and ATTR after adjusting for all known predictors. And this is a summary of the diagnostic accuracy of the main parameters we discussed, LGE, NATT1, and ECV. And you will see that probably is the ECV that has got the highest accuracy. And so what we do with CMR, using T1, ECV, and LGAD, we can really image the natural history of amyloid deposition, going from patients with no deposits at all, from very early deposits, more deposits. And the beauty of it is that all this information is co-registered with all the information also on structure and function. So you can follow really the natural history of amyloid deposition and all the function, structure, and sequences. But so going back to the patient from before, I said from this image, we cannot tell if the patient is better or worse. But if now I show you the ECV maps, you can see that in 3 years, there was a significant progression. The myocardium went from green to red. And if you simply draw a region of interest, you can see that in 3 years, the patient went from an extracellular volume of 45% to an extracellular volume of 67%, so very significant progression. But then Does tracking changes over time has got any clinical meaning? This is a study recently published, and what we found here is that any change in ECV in patients with AL amyloidosis undergoing chemotherapy, was a cohort of more than 170 patients, so a change in ECV at 6 months and 1 year independently correlated with prognosis after adjusting for the known predictors. And why is that? Why measuring the amyloid in the myocardium matters? Because amyloid is a very dynamic process and is the final common pathway of two main mechanisms. The amyloid production that we can measure with free light chains and then with TTR levels, but then also there's a natural clearance in each individual, but which is extremely variable in the different individuals and we cannot measure. But by measuring the cardiac amyloid load and also in the liver and the spleen, what we do, we measure the final common pathway of all these mechanisms that determine the amount of cardiac amyloid load and therefore the prognosis. So measuring the myocardial substrate really matters. So current available evidence is that LGE has got a very high accuracy, and so it's very useful diagnostically, but cannot be used to track changes over time. And this is where ECV mapping really has got an important role because of the high precision. So when we think, so which of the surrogacy aspects do we know are met for this modality for AL and TTR? First of all, I would like to say that it's still very early days because we have got only very few studies. But if we think about statistical significant drug-reduced movement of the image biomarker, so ECV, we've got one study in AL, 170 patients. ECV, we've got fewer, smaller studies, but significantly smaller. When we think about statistically significant drug-induced movement, clinical benefit, again, ECV in AL amyloidosis, we've got one study, but it's ECV only cardiac, so we don't have anything yet on the liver and the spleen. Image biomarker is prognostic at baseline. There are several studies that prove that both LG and ECV are important independent prognostic marker, both in ATTR and AL. And also that the majority of effect of drug-induced movement clinical outcome is attributable to the drug-induced movement. Again, very early days and probably in one study with ECV in AL amyloidosis. So thank you very much for the attention and looking forward to the discussion.
Thank you, Marianna. I now invite Dr. Ruberg to lead the moderated discussion.
Thank you very much, Dr. Dorbala. Thank you very much, Marianna, for that wonderful presentation. It's really a pleasure and an honor to be here. I'd like to invite my co-panelists to turn their microphone on and participate. So Dr. Fontana well characterized and reviewed the basics of MR and the utility in amyloidosis. I do think it's important, though, for us to think a little bit practically as well, and those would be the limitations of the modality in respect to patients participating in clinical trials. Number one, patients with implanted devices really are challenging to image by CMR. It can be done with certain techniques, but those techniques aren't necessarily broadly based. Second, patients with impaired renal function are often excluded from contrast-enhanced MR, although with the new macrocyclic agents, the concern about nephrogenic systemic fibrosis is less. And third, unlike echocardiography, the MRI exam is not easy for patients. It's 45 minutes long, typically, or longer, and requires multiple breath holds. And for sick patients, that can be challenging. So I think these are important considerations as we think about the modality, which we're about to, I think, boost and say how great it is. But that we have to also have, kind of clear eyes about how patients actually experience these tests that we do. So I'd like to start actually to give appropriate time to non-ECV metrics, because we're going to spend a lot of time. We have 18 minutes. Most of it's going to be about ECV, but I want to make sure we do talk about other things that MR can do, because MR, since the image quality when patients can complete the exam is typically outstanding, with excellent edge detection, the volumetric calculations tend to be more accurate and reproducible than echocardiography. So what about LV mass? What about and mass regression, which has been reported in MR using certainly silencing agents in a small study? And what about quantification of late enhancement using the various techniques? So maybe I'll start with Justin, who's sitting to my right.
So I do imaging research, and the caveat is I myself am not an imager, but I know a lot about these epidemiologically, and I think certainly I can't answer that question specifically, but I think certainly one of the advantages of MR is you get, you know, as close to gold standard cardiac morphology and function assessments as possible. But I think you cannot understate, as somebody that does imaging research, just the practicality of, again, not to be negative because I'm very pro MRI, but of just getting in the scanner and, you know, putting your study participant through that assessment. So it's clearly not as widespread as, you know, for example, you know, doing a strain echo at the Brigham Cardiology Clinic.
Maria, is your hand up, Marianna? Did you want to speak?
Yes, I just want to do kind of more general comments. I think we are talking about endpoints, but in a way, phase 1, phase 2, phase 3 imaging endpoints are actually a very, very different concept in the sense I find that certain imaging modalities and certain studies are very suited for a phase 1, phase 2, and actually not for a phase 3 in the sense. And so I think that when we talk about these different imaging modalities, they might have a more prominent or less prominent role depending on what phase of the trial we are. And so, for example, I think that cardiac MRI, I think, is a beautiful technique, but a bit biased, I guess, because really you can image every step of the physiological pathway which leads to amyloid deposition from the deposit to the function to the structure and everything. And I think that is very well suited for example, phase 1/2, much more complicated for a phase 3, where it should really only be used in sub-studies. Echo almost is the opposite in the sense, echo is suited very well from phase 1 to phase 3, and in phase 3, you don't need really an echo sub-study because it's so widely available and cheap that can be done in all patients. So I think some, in the sense, we need really to put into the context of the phase of the trial that we are talking about.
Yeah, I think Marianna makes a really key comment there, you know, and I think, and this dovetails with what Rick was talking about as well, but if we think about the richness of the information that we get with MRI, and I think there was a question raised about echocardiography is what else should we be looking at? And I think MRI gives you the advantage because of a lot of its strengths that Rick was elucidating, that maybe there is something we can learn that we didn't think we should have known. You know, an MRI at least gives us the capacity to do that in a highly sensitive way.
Thank you, Justin. Excellent point. So I'd like to now move to discuss ECV in particular, because that's really the kind of the principal parameter I think that would be proposed for use as a surrogate. And I think ECV kind of suffers in some ways from the challenge of reproducibility. And so I thought I'd ask Michael, looking at him, to as our MR physics representative, to speak a little bit about inter-vendor reproducibility, and talk a little bit about, educate us about the challenges that CMR ECV faces in terms of reproducibility.
Yeah, thank you, Rick. I mean, ECV has made a big impact because it is related to the amyloid load, but it does suffer, as you say, from technical issues. So it is based basically on measuring the so-called T1 relaxation time before contrast and after contrast. And in particular, the T1 time before contrast is, for example, heart rate dependent. So if you have a patient with a slow heart rate, you can potentially get a different value than if there's a fast heart rate. And this in some way, even though this has been minimized, probably is still bedeviling some of the data which we get. And then the other thing, actually, it was mentioned, we have a variety of contrast agents. We have the macrocyclic contrast agents. Some of them have affinity to proteins, and there's some degree of binding. And so the ECV, actually, the very concept when it was introduced treated the contrast agent as something totally passive, and there was an equilibrium between the extracellular and the vascular space. And if there's some protein binding using different contrast agents, you can potentially get different results. Now, I don't want to paint an overly negative picture because overall, ECV, for example, does have the advantage that if you do a 3 Tesla scanner or 1.5 Tesla scanner, or your patient with a device goes even to a half Tesla scanner, that in principle you can compare ECV, which is not true for the underlying relaxation times. But we do have these technical issues, and if we're thinking about multi-center trials, that is a considerable challenge. We have done some work at the Brigham in the mavacamten trial, where we also used T1 and ECV. It wasn't a primary endpoint, but we could see some of the issues which increased the variability. So I think there's still work to do. And then finally, the theory behind ECV is fairly simple, but you know, in terms of the biology, it's probably more complex. So what I'm getting at is ECV, for example, during disease progression may have a different meaning to from ECV in a patient which has gone into remission. And I think that's also important, that you have a surrogate biomarker whose meaning may actually change, and I think that may be an additional challenge in this.
I'll ask Dr. Cuddy actually to elaborate on that point actually, because I know she's published on that with Dr. Dorbala. Thank you, Michael.
Yeah, I published on that with a lot of help from Michael to understand, because it was cross-sectional. So unlike the data that Marianna has presented, it wasn't longitudinal that we published on. So it was a cross-sectional cohort of patients with active disease who still had elevated free light chains versus those who were fully in a hematological remission. And we were surprised that the ECV was fairly comparable between the two groups. So even though these patients in remission had survived longer than a year, had done well with treatment, we hadn't seen a massive shift in their ECV. So it was still like we equated it to maybe the amyloid still being there. But then the other questions, as Michael alluded to, were, is there something else happening within the myocardium in remission? Is there a fibrotic process that's replacing the amyloid that's being removed? So I think the ECV gives us definite kind of information, but I don't know if we fully understand what those changes, exactly what Michael said. And one other point is, I've sat and drawn hundreds and hundreds, if not thousands, of circles on these maps. And there's a lot of heterogeneity. You know, you see it, and like it's kind of fascinating to me, because I'll see one patient, their ECV will be really high in the basal septum. It'll be in the 70s, but in the lateral wall, it'll be in the 40s. As I go more apically, it'll be in the 40s. So, like, where do you draw your region of interest? How many slices do you take? How do you reduce that variability when there's already heterogeneity throughout the myocardium? I think that's a huge challenge with ECV.
These are great points. I'm going to ask Dr. Fontana's response to this too, because I know that she has perspective. But I wanted to ask Michaela for the experience of actually incorporating ECV into a clinical trial.
Yeah, so we are doing a very ambitious MRI sub-study. We are looking for 150 patients, and will be a multicentric study. So we are facing all the challenges that Michael mentioned, but to overcome these challenges, we have a very rigorous protocol. So we have chosen labs with similar scanner. Of course, we are having the same protocol to scan the patient, and using the same contrast. So when you, and more important, we have a core lab that is led by Dr. Fontana. So when you try to control for all these factors, I think the quality of the study increases massively. And so we're very enthusiastic and excited for this sub-study, because also we will be able to correlate the MRI, the richness of the data we will get from MRI with other imaging modalities like echo and scintigraphy, and also with outcome. And more important, we will learn a lot about the silencer effect on the, not only on ECV, but also in the function and the structure. So.
Right. Excellent. Yeah, I think you made so many important points about actually how this has to be done right. The core lab, unifying the vendor techniques. We didn't even talk about that there are a couple of different ways to actually acquire T1 data, and there are differences between vendors, and they are not necessarily.
Yeah, so we're doing very good, and Marianna, their core lab is checking also for the quality. So far the choice of the labs has been done right because most of these studies are accepted.
Thank you. So I wanted to ask Marianna. So I thought Sarah brought up a really good point, and I'm thinking about it in contrast to ECHO. So ECHO, we have two basically measures just to review, GLS and apical to basal strain ratio. It's been reported. CMR, where do we take this ECV measure? Does it have to be a global ECV from every myocardial segment, or does it have to be from the basal septum, as you have reported before? What's the most reproducible? And if we're going to use this as a and you're going to be using it as a marker, what's the best way to do it? And you're on mute, I think. It wouldn't be a Zoom meeting if you were.
It took me kind of 11 years, and I do report on average 20 AMLET scans per week to understand that actually the simplest approach is the more robust and especially precise to track changes over time. Because as Sarah was saying, there's a very heterogeneity, especially with the gradient from base to apex. So what happens when you take the 3 short axes and you want to do whole RTCV is that if 6 months after you are 1 centimeter lower or 1 centimeter higher, actually the ECV measurement changes significantly. And so what I think is the best approach, and that's what we use to in tracking changes over time in both type of amyloidosis now, is 4-chamber, a single regional interest in the basal to mid septum. And this has got both a technical advantage, but Michael, I'm not going to comment on the technicalities of this, but basically our physicist Peter Kalman always says that on the intralateral wall there are lots of reasons why we get much more artifact than in the septum. So from a strictly technical point of view, the septum is the best one with the best signal-to-noise ratio, but also with the 4-chamber, because you plan it off 3 points here, more or less always in the same place. So single region of interest in the basal mid-septum of the septum, we think that is the best way to track changes over time, because you are almost always roughly in the same location, so you really increase the precision. It may be less sensitive because what we've clearly seen that when there is regression, we've got many data from AL, the regression almost always started from the lateral wall. So you lose a bit of signal, but you definitely gain in precision. And the fact that a single region interest is the way to go, it doesn't surprise me that much if we think about the T2 star. T2 star is the only example in MR whereby an imaging and a single measurement guided treatment response, and by doing so, changed the patient prognosis. And with the T2 star, we do exactly that, a single regional interest in the septum. So I'm really in favor of a simple and reliable way rather than trying to complicate our lives and doing whole art where actually we lose some precision.
Thank you, Marianna. Ahmed, Dr. Masri.
Thank you, Rick. A couple of quick points. One is a lot of the issues with MR, once we are doing within the same patient serial measurement in a clinical trial and as an endpoint, a lot of these issues of variability across patients go away because we can use the same exact protocol, the same exact location. We can prescribe the same exact, sequence, position of the slides, everything. So a lot of these issues go away once you are doing at the patient level, not using it as a cross-sectional across different individuals. The second thing is we want everything from MR, so we're really greedy. When we talk about 90-minute protocol or 120-minute protocol, because we want to get everything. But once you're in a clinical trial and you want an individual endpoint or 2 endpoints or 3 endpoints, whatever you want to focus on, it can be as short as 10-15 minutes. In oncology, this had been done because of strain reproducibility issues. It's been done in oncology where you do 2, 3, 4-chamber views. You can do that in 5-6 minutes and do post-processing of the strain later on if strain is your goal. If you want to do, for example, ECV, you acquire T1 native, you give contrast, you acquire post. And so there are ways of dealing with this in a different fashion that can be used in clinical trials.
Those are great points. Thank you for making them. As we get better scan technology and faster acceleration, scan times do get shorter, and you can do targeted scanning. So I think that that's critical. I also wanted to make the point that ECV, we talked about turning off production. That's how we treat the light chain and how we treat the prealbumin protein. But and generally we look at kind of slight changes in the interstitium that Marianna already talked about that have to do with kind of the steady state of reabsorption. But we are developing not I, but we around this table are developing agents that might remove amyloid from deposited tissue, in particular the heart, and have already been others have been tested. And theoretically, ECV affords a means to actually resolve the change in the interstitium that would occur in a way that potentially other modalities, with the exception of maybe PET imaging, don't. And so I thought maybe we could throw that open to a quick discussion in the 2 minutes and 11 seconds we have left.
I have one thing. I think Ahmad's point can't be understated, and I think as we think about, the whole goal of this is that we need to be forward-thinking. And I think this is important for echo and in particular for MRI and perhaps other imaging modalities, but the influence of serial imaging. So not only from the per patient variability could perhaps be reduced, but it also enhances power to make an observation. So, we're here trying to, figure out how is disease changing, and certainly you can have smaller patient sample if you have repeated assessments. And so it might give us higher resolution to truly define or truly see, or the capacity to see a difference. So as we think about moving forward, is it enough to have a pre and post, or should there be something serial in between? And that will reduce the likelihood of getting perhaps a false positive or maybe even a false negative.
Thank you, Justin. Spencer, you wanted to make a point?
Yeah, thanks, Rick. And thanks for bringing up the discussion about thinking about antifibril therapy. So working as a drug developer who works on antifibril therapies, just to ask a provocative question. So we would obviously, and I think most of the antifibril companies are now using ECV in their trials. If ECV decreases, I think we would all be very happy. But if ECV doesn't decrease, does that mean that the drug didn't remove amyloid?
That is a very good question, and we have 40 seconds to answer that, Michael.
So I think one exciting development in MR is actually using artificial intelligence for the actual scan procedure. So there is one company at least which is automating the whole planning procedure in cardiac MR and is reducing the scan time, or can reduce it quite drastically. We're using it even clinically, and the variability and quality of the studies has been reduced significantly. So I think that's an exciting development, which for multi-center trials could actually be a game changer.
Right, and as well as virtual late enhancement. I think you just wrote an editorial in Circulation on that, as I just read 2 days ago. So I think I would love to answer Spencer's question, but I think it'll probably happen in the Q&A, and I think Sharmila wants to move on.
So thank you so much. And we are out of time now, but we'll definitely come back to Spencer's question during the moderated discussion session. So we are really grateful and thankful to all of you for really an amazing discussion. We now have a 10-minute break, and we reconvene at 10:35 a.m. here. Thank you. Hello, can everyone get back to your seats, please? We are ready to start the session. So, I think we can just Preston was saying. Yes, sir. Hi all, could you please have a seat? We want to get started. It's 10:35. I think Kristen was.
So thanks for coming back in. We're going to get started with a session on scintigraphy, and I just wanted to take a moment to sadly recognize one of our esteemed colleagues, Claudio Rapezzi, who really is a genius. Those of you who knew him, work with him. He's not only lovely, but quite curious. He was I would say, a master of medicine who knew medical history, besides just in medical facts. And as we know, believe it or not, in 2005, which is nearly 20 years ago, he was the first to describe the use of scintigraphy and develop the Perugini score. Kind of amazing it took us this long to get to this point, but Claudio was ahead of his time. I had the privilege of co-chairing the ATTR ACT trial with him and learned a tremendous amount. And anyone who knew him would remember what I think I colloquially term Rapezziisms. These little comments, and I think Michelle, who's on the call, wrote this in a recent testimonial, which is he was kind of supernatural in his capacity for synthesis. He made these lightning associations and had vertical intelligence and extremely sharp irony. So we'll miss him. I'm sure he'd be proud to be here with us, and he'd be very glad to know that we're trying to move the field forward for all of our patients.
Thank you very much, Dr. Maurer. And this talk was originally planned to be delivered by Dr. Rapezzi, and here I am filling in for his shoes. And we all miss him, but we've learned a lot from Dr. Rapezzi. So this is me presenting on behalf of the panelists. Dr. Omar Abu-Azzeddin is online. Dr. Maurer, Dr. Soman, Dr. Sperry, and Dr. John West are here with us. So pyrophosphate imaging is not new. This razor was developed in the early 70s to image myocardial infarction, and it was recognized early on. This was published in '82, that myocardial uptake, especially when it's diffuse associated with thick ventricles, was indicative of cardiac amyloidosis. Then here's one major study led by Dr. Gilmore with participation from several of the panelists here, which included a large number of patients, nearly 1,500 patients and nearly 400 with endomyocardial biopsy. And in this study, 3 different tracers were used, pyrophosphate, DPD, and HMDP. And in select patients, grade 2 or grade 3 uptake of these tracers, if AL amyloidosis was excluded, was nearly 100% specific for ATTR cardiac amyloidosis. Again, the importance of this finding is really significant because it has changed the disease a lot. So could you just, next please? Sorry. So this image shows you what is a grade 2 or grade 3 visual PYP uptake. On the top image, you see uptake transaxial image of the heart, and all the colored stuff is PYP activity in the myocardium, left ventricle and right ventricle. That's almost as equal to rib uptake, and that would be grade 2. And the bottom, where myocardial activity is greater than bone activity, that would be grade 3. So this is what led to an entire transformation in this field and led to development of guidelines which now say that this could be used for non-invasive diagnosis of imaging transthyretin cardiac amyloidosis. So can we go next, please? So experts got together and developed consensus recommendations for diagnosis of AL and ATTR cardiac amyloidosis, and these are the recommendations. You need either endomyocardial biopsy proof with typing of amyloid or extracardiac biopsy proof of amyloid plus typical cardiac imaging features to make a diagnosis of AL or ATTR, or abnormal biomarkers to make a diagnosis of AL amyloidosis. The bottom piece of the panel is what is most interesting for us at this point, which is based on the data from Dr. Gilmore and all others, we now know that we are able to make a diagnosis without biopsy in a select group of patients, particularly those who have typical cardiac imaging features or echo or MRI, those who have heart failure, in whom a clonal plasma cell process is excluded. If you see grade 2 or grade 3 pyrophosphate, DPD or HMDP uptake in the myocardium, that patient is diagnosed with ATTR cardiac amyloidosis, and we don't need biopsy in that patient. Next, please. So in fact, in this registry, the THEOS registry, a large number of centers enrolled participants into this study, and we showed that as of 2016, imaging-based diagnosis of amyloidosis has exceeded tissue-based diagnosis of amyloidosis. So this has really permeated into clinical practice. Scintigraphy with PYP and DPD in the pre-treatment era, before we had therapies for TTR amyloid, was prognostic. And here you have data from PYP on the left, from the Columbia group, DPD in the middle from the Dr. Rapace's group, and DPD on the right from the National Amyloid Center, from Dr. Wechalekar, Dr. Fontana, Dr. Gilmore, and others. So myocardial uptake defined in the first case as heart to contralateral ratio more than 1.6 was associated with worse survival compared to patients who had a heart to contralateral ratio less than 1.6. Similar thing with DPD, and similar thing with visual grades, which is the right side of the panel, where grade 0, no myocardial uptake patients had better survival compared to grades 1, 2, or 3, which is patients who had myocardial uptake. Now, emerging data that was presented by Dr. Matt Maurer, actually the ISA from the Alnylam study, really looked at an exploratory endpoint looking at reduction in cardiac amyloid burden with technetium-labeled scintigraphy, all tracers, compared to placebo after 12 months of treatment with patisiran. So in this graph, you can see in blue is the treated patients, and in magenta color is the placebo group. On the bar graph, you see that the patisiran group had either no change or reduced visual grade, whereas the placebo group had no change and one patient had increased visual grade. And this is not surprising. Without therapy, there was no change in visual grade, and with therapy, either there's no change or there was a reduction in 30% of these patients. And this is really very interesting finding, because this is for the first time we are now able to show an improvement in visual bone avid tracer scintigraphy using targeted TTR therapy. And we know from cancer literature, evidence using FDG-PET, that molecular changes may foretell eventual improvement in cancer burden. So these findings are not surprising. What we are curious to know is, does this improvement really tell us something about future improvement in these patients. But what we don't understand is really the mechanism of PYP uptake. So why is PYP taken up by the myocardium? What does this change mean? So although we are all very optimistic, we need to step back and question, does this represent some kind of altered fibril binding characteristics? Is it a growing fibril now no longer growing? And is that sufficient to change this signal? Is there onset of myocardial edema, fibrosis, cellular infiltrate, anything else that could explain a reduction in this visual grade? So this is something that we need to study more. More importantly, you'll see that on the right side, a number of patients remained at grade 3 from baseline and after treatment, even with patisiran therapy shown in blue. The question is visual grade a good enough test? Do we need quantitation? And now we are able to do quantitation using SUV-type metrics with SPECT-CT-based technology. And the question is, if you can quantify myocardial uptake using standardized uptake value, and then look at this as a metric, could this provide us more information than visual grade? Could this tell us information about early change? Could this tell us information about response to therapy or early detection of disease even? And these are the questions that we still need to study and answer. So I just want to conclude with one or two slides saying, really, bone avid tracer scintigraphy has transformed this field of cardiac amyloidosis. If you think about 30 or more years ago, we thought ATTR amyloidosis is rare, it's highly fatal, and you really needed a biopsy to make a diagnosis. Now we know that biopsy can be avoided, and we can make this diagnosis non-invasively. We had no treatments for this disease until as recently as 4 or 5 years ago, and we were primarily treating heart failure. But we now have many approved therapies for treatment of TTR amyloidosis. Patients who were diagnosed by biopsy were eligible to participate in these clinical trials. Think of the significance of this. If you needed biopsy to put a patient in a treatment trial, these trials would not have been completed. I think it has such a transformational effect that patients could enter the trials based on an imaging-based diagnosis that led to approval of several therapies, and now patients diagnosed can benefit from treatment of their disease based on imaging. And this has really been really hugely helpful for the field. More importantly, we are now knowing more about the epidemiology of this disease. We are able to perform screening studies using this non-invasive approach. We now recognize that the disease is much more prevalent than what was previously understood or known. So I think this is something we need to keep in mind. So to conclude, what's the current evidence and future directions? This is really a robust marker. Bone avid tracer scintigraphy is a robust marker to diagnose cardiac ATTR amyloidosis. It provides prognostic value based on baseline values. But what are the gaps? I think the mechanism of tracer uptake and the mechanism of host therapy changes are poorly understood. We need really more studies on quantitation of this. Which surrogacy aspects does it mention? Statistically significant drug-induced movement of biomarker. Yes, some preliminary data that I just showed you. Statistically significant drug-induced benefit of clinical outcomes. We have multiple drugs now for this disease. Imaging biomarker is prognostic at baseline. Yes. I think the last component where majority of the drug-induced movement of clinical outcome is attributable to the drug-induced movement of biomarker. I think this is where we are lacking information. So thank you very much.
Thank you, Sharmila. That was fantastic. We have a quick panel discussion. I wanted to open it up to some of my colleagues. Maybe I'll start with Omar, who's online. But I wanted to ask each of you at your centers, how is scintigraphy being utilized? Are you using it for diagnosis, for prognosis, for monitoring of therapy? What's the current lay of the land at your center?
Thanks, Matt, and thanks, Dr. Dorbala, for that great presentation. So currently at Mayo, we are, I would say predominantly the use is for diagnosis. in some patients who have had therapy through clinical trials, they have had serial studies, and so we have a very small number of patients with serial imaging, but really it comes down to diagnosis for us here. Prognostication, there's equipoise on that. The Columbia group, Mara's group, has shown the H/CL ratio was prognostic. There's been other data from Cleveland Clinic which was somewhat discordant with that, but I think it goes back to what Dr. Dorbala said as far as quantification. I think that is where I think the H/CL ratio is very crude, and I think better quantification of myocardial uptake may indeed be prognostic, but more data is needed for quantification. I'll just pass it on to my colleagues.
Thanks, Brett. Thanks, Matt. Totally agree, Omar. We're using it the same way in Kansas City. Diagnosis mostly. I just want to add that our patients that have hereditary gene mutations but don't necessarily have cardiac involvement, we scan those at some interval, which I think is not completely clear what that interval should be at this point. We've scanned a few patients serially who've been on combination therapy and have seen some nice reduction or removal of PYP agent. And speaking to Matt's point earlier, we don't really know what it's binding to, I think, at this point, so we don't know what that means for outcomes. Prognosis-wise, totally agree. I think HCl ratio, I think as we're learning more and more about this, is becoming more of a crude measure, and hopefully as we use more SPECT CT, SUV type measurements, amyloid burden type measurements with the PYP, we can actually follow changes more easily over time.
Prem, anything to add at your center? How's it being used?
We use it mostly for diagnosis, but I just want to say that it really is an exceptional diagnostic biomarker. And not just because of the diagnostic accuracy numbers, which are sort of very unusual in the imaging world to get 100% specificity, but also because it can be used in anybody, irrespective of body habitus, irrespective of implanted devices, irrespective of renal function, contrast, allergy. So if you order a PYP, they get it. And as far as nuclear techniques go, it's a relatively low radiation dose. I think it's about 3 millisieverts, which, to remind everybody, is the annual background radiation we get for the privilege of living in the United States. So if the prognostic use, I think, is hampered by two large gaps in our knowledge. One is we don't know what it binds. That's a really important area of knowledge gap. The second, I think, is in TTR, we don't really know what happens in the myocardium when you treat patients. And unless you have these two bits of information, it's very difficult to use this tracer as a prognostic biomarker in this disease. Although I don't know if patterns and intensity of uptake at baseline, we're talking of two different prognostic uses. One is, does the intensity of uptake and the pattern of uptake, for example, RV uptake, does that have a prognostic implication? I think serial changes is a tall order with PYP, is my opinion. Thank you.
Yeah, I would echo, we basically use it for diagnosis and not for prognosis or monitoring disease, despite whatever data we've promulgated. Can we turn our attention just for a minute to this issue, which I don't think we've touched upon in any of these imaging modalities, and that is, we're all at expert centers, but what does it look like in your experience in the community? And maybe we can then beat up on the HCl ratio, because it seems to lead to a lot of false positives. So I don't know who wants to start, but experiences at your center looking at what's happening in the real world, so to speak, outside of the pearly gates.
I
guess I'll start.
Oh, go ahead.
Yeah, go ahead, Omar.
Oh, I was just going to say two of the biggest issues that we've run into that we're seeing in the community is planar only imaging. So you could see what looks like positive uptake on planar only imaging. But indeed, when you do SPECT imaging, which is absolutely necessary, as the consensus documents have stated, it's all blood pool. And it's not myocardial in nature. That's one. And then the second, I think, Matt, you have very nicely published upon the need for ruling out light chain amyloid, because remember what Dr. Dorbala said, you need to have ruled out a monoclonal process for this modality to have 100% positive predictive value. And so in the community, we are seeing that that isn't always being done. And so I think that's one of the most important things, if I can highlight, that we have seen as a center, and I'm sure others can comment. Sorry to go ahead.
No problem. Anyone else?
I'll just echo that. I think those are the exact things that we're seeing too, and we've all discussed at other venues as well. These are the things that we're seeing. I think when we're talking about using any of these three imaging modalities, echo, MRI, or PYP or bone scintigraphy, what happens in a clinical trial versus what happens in the community can be very different as far as image quality, and definitely PYP is not unique in that area. I think we've taken an approach in Kansas City of just directly communicating with the other centers that are doing these planar only images, or are taking the SPECT images but aren't actually looking at them when they're doing the interpretation, to work with their nuclear labs and their nuclear technologists on how to get the right protocol, how to look at the guidelines that Sharmila and others here have published, and do it according to those guidelines.
I just want to add, from an acquisition perspective, it's the easiest thing to teach people. So when we started, we had a lot of issues in the community. We have a lot of rural centers. People don't want to travel 5 hours just for an imaging test. And so just by giving a phone call, going over the scan, what should be done, how it should be done, right now we haven't had any issues for more than a year and a half compared to when we started. And so it is even easier than echo when you're giving feedback to people on how they can improve the scanning technique.
Dr. Mara, it looks like Marianna has a question.
Yeah, sure.
Sorry.
I didn't see it. And maybe, Sarah, if you can let us know if anyone's online, too. Yeah. Marianna, you want to comment?
I just wanted to make a comment. I mean, I think what DPD has done, what bone scintigraphy has done in cardiac amyloidosis is a complete revolution, in the sense there's absolutely nothing imaging-wise that is so sensitive and specific and actually allows us to make a diagnosis without a biopsy. And for that purpose, I think it doesn't even matter too much that we don't understand the mechanism. It works, and it works beautifully, and for diagnosis, and that's all we want. I think much more careful we need to be about tracking changes over time, because when we give the tracer, and sorry to all nuclear medicine nuclear medicine physician on the call for my very simplistic approach to this, but in the sense that when we give the trace, there is a lot of competition with other compartments that are much larger than the heart, because the heart is only 300-400 grams, and instead the muscles is a huge compartment, soft tissue, bones, and everything. So any change in any of those compartments will also determine a change in the cardiac uptake, and we should be extremely careful in attributing reduction in the cardiac uptake to reduction actually in the cardiac amyloid load. And we have quite a lot of experience with the follow-up, and what we see is that we see significant reduction when we use silencers or stabilizer, but like huge reduction with actually no change in myocardial structure and function. And so you start to see a discrepancy between the cardiac uptake and actually what happens to structure and function of the heart. So do we, I think, really understand what reduction in the cardiac uptake means? Probably not. We know that there is a biological effect at the body level, but I think we should be very careful in saying that is a reduction in the cardiac amyloid load.
Cristina, you want to make a comment?
So first of all, I agree with Marianna, and for the purpose of clinical studies, I think that understanding the effect of a drug to the patient as a whole is extremely important, especially when we think about the fact that patients have extra cardiac involvement, even if it's not primarily the cause of the patient's sickness. But this is definitely a beautiful diagnostic tool. However, when we think about implementing it in the context of clinical trials, how should we think when we have different modalities? For example, how do HMDP, PYP, and DPD perform? Should we think about having certain protocols across different sites around the world? Is it exactly the same? I'm just asking because when we think about cardiac MRI, we basically have narrowed down to one single agent, one specific protocol, one almost type of software. How does it work with bone tracers, which is, of course, more widely available even in local centers? How should local centers use this when it's PYP or DPD? And the region of interest is also different, right? When you trace it between the different modalities.
I think that's probably an incredibly important and unanswered question. I turn to Dr. Vest for a minute. Just obviously there's this emerging provocative data from silencers that the scans get better. Maybe you can comment a little bit about that, and then maybe Christina's question in the approach as a sponsor. How are you trying to think about different agents, different communities making imaging, and to include this in clinical trials?
Yeah, thanks so much, Matt. So we have now used this in multiple international studies with both the Apollo B data of patisiran, which Dr. Dorbala shared in her presentation, as well as the Helios A study of vutrisiran in patients with hereditary ATTR amyloidosis. And we have had what I would characterize as very consistent observations across those studies, and certainly something that we've been excited about. In terms of employing this in a clinical trial, I think this probably falls somewhere between echo, which we can use at every single site in a large study, and MRI, which is very complicated to use in a large study. We've certainly been very selective with technetium about our sites. This is not something that we've used at every single site, and we have chosen centers that we believe are going to provide high quality results. Use central reader for analysis of the data, and I certainly think that's very, very important. The question about multiple tracers, also an important one. In international studies like this, we've certainly allowed different tracers to be used. Whether or not that needs to be more standardized, I guess that's a point for discussion. I think that would certainly greatly complicate the ability to use this in a large international study. But we're certainly very encouraged with our experience today.
And just to follow up on that, I mean, I think the final question I want to ask everyone is, is PYP ready to actually track disease, monitor disease in the context of a drug therapy? My own opinion is absolutely not. I'll say there are a lot of unanswered questions. But if someone thinks it is, feel free. If you don't, like I do, I guess we could try to brainstorm what are the pathways forward. And I think your question about these three isotopes, they look in Julian's paper like they all perform equally well, but it's, I think, a little embarrassing at this point that we haven't done a study in the same patients really figuring out whether these isotopes are equal in their sensitivity, specificity, and so forth. And that may, I strongly believe, need to be done before we embark on trying to, monitor disease progression with three different isotopes. But
yeah, so I just wanted to extend on that, which is the soft tissue uptake is significant with DPD. not with PYP. And one of the questions that I think John West and others could do is look at their data and separate out to see if the improvement, are you seeing it primarily in the DPD cohort, or are you seeing it in both DPD and PYP cohort? And that can give us some answers to what Marianna raised, a very important point about is it reflecting decrease in the heart or is it a relative decrease in the heart, because there's an absolute decrease in the whole body tracer activity or competition from other sites. I think that could be one area.
Gotcha.
So one last question I had was...
So I can answer a little bit to that, is that we actually looked at the compartment transition of DPD in the bones, in the muscle and the heart. And we actually imaged patients as long as we could get them. So we got them in at 3 hours, 6 hours, 24 hours, and 48 hours to see how the tracer redistributes. And there is a redistribution of the tracer from the heart and the soft tissue to the bone over time, till we can't image the patient anymore. So I think it's really important to remember that this tracer is not just taken up by the heart, it is taken up by the bone and by other tissues. And there is a redistribution between the different organs as the tracer probably comes off the heart and goes to the bone. So what we have not understood and what we have not done is, because we've all imaged patients at one time point with PYP and DPD, we don't know what happens after treatment. Do we change the redistribution in some way, which is what's affecting what you're seeing in the different organs by what you're doing to the amyloid? And I think we need more studies of not just a single time point imaging, but serial time point imaging to understand what these tracers are doing.
If I can add one more comment, Dr. Mara, real quick, I know we're approaching time. As you know, we're trying to push early diagnosis because we think these drugs work better the earlier you diagnose. One of the important studies that needs to be done is using perhaps PET imaging or whatnot to see what is the burden of amyloid that is necessary to start seeing a positive PYP scan. Yes, the modality is brilliant, and I think it's really revolutionized the field, as we've all stated. But these are in heart failure patients typically. Are we missing an opportunity in patients who don't have as high a burden, perhaps, to have a positive PYP scan? And that's something that I think we need to take advantage of hybrid imaging to answer.
Brett, anything to add?
I agree with the points, particularly about the different tracers being all very different. We had a shortage of PYP recently this year, and a lot of our centers used HMDP, and anecdotally we found that HMDP seems to bind stronger and have the negative studies are seemingly more negative with less blood pool. And I totally couldn't agree more with Ashu's point earlier. I think imaging people at multiple time points is really important, and not just starting at 1 hour. I really think that this tracer gets into the heart very early, within 5 or 10 minutes, and then we're probably going to see it wash out a lot faster as patients are treated or patients improve.
Thanks all. That was great discussion. I appreciate it.
Absolutely. Excellent discussion, and nice lead into the next topic on molecular imaging of PET. Oh, sorry. These are all the panelists. I'm presenting on behalf of Dr. Michele Emden, who's online, Spencer Guthrie, Dr. Jen Sorensen, Dr. Andrew Stevens, and Dr. Jonathan Wall. All of them are here. So if you think of positron emission tomography, really molecular imaging based on PET identifies changes in physiology and metabolism well before you see an overt change in anatomy or function. This is true for any type of disease, including cancers. The biggest strength of PET-based molecular imaging is really you have targeted molecular tracers that can bind to a certain process, in this case amyloid. They have very high sensitivity, high temporal spatial resolution, and the technology lends itself beautifully to absolute quantitation. And hybrid imaging allows us to study the systemic disease in a much better fashion than we could have done without hybrid imaging. So when you think of beta amyloid PET tracers, they were originally developed analogous to thioflavin T. This is a dye that binds to amyloid, and this appears to bind to the binding sites on the beta-pleated sheet structure. So if you look at fluor-beta-pair, flutimidamol, fluor-beta-ben, or C11 B compound, all of them seem to show uptake in amyloid independent of the precursor protein. The other new compound that we and others are working with is labeled with I-124. It's P5 plus 14. It's a synthetic peptide that appears to bind to highly electronegative surfaces, such as heparin sulfate, heparin, and amyloid fibrils itself. And whether it is the beta amyloid binding tracers or this novel peptide, these seem to provide an image of amyloid. And here's just an example that these tracers provide not only an image of the heart, but an image of the whole body. And this could be a huge advantage for systemic diseases such as AL amyloidosis particularly, and probably ATTR, where lung may be also affected. So here's an example where you see cardiac uptake and pulmonary uptake, and on the right side you see pulmonary uptake. Now contrast that with a control subject who's enrolled into the study who has no myocardial uptake, and you see the liver activity is physiological excretion of this radio tracer. Now a different tracer, you can see imaging of ATTR heart, ApoA4, as well as AL heart. And in the AL, you can see liver activity, spleen activity, as well as kidney activity. Again, this is I-124 P5 plus 14, a different tracer, but again, giving us information about cardiac as well as extracardiac. Now, look at controls. This is just physiological activity in the stomach, but really nothing in the heart at all or in the other organs. So a number of investigators have looked at this. Dr. Sorensen, who's online, his group published initially in 2013 with C11 B compound showing that myocardial retention of this tracer in amyloid patients shown in red with 95% confidence intervals is substantially higher than in healthy controls shown in blue with pretty much no overlap at all. Then this is our data using fluor-beta-pair. Again, 95% confidence intervals not overlapping. This is using fluor-beta-pair in controls compared to amyloid. Then the third study is from Dr. Michele Emden's group from Italy. They looked at fluor-beta-ben. Again, you see in blue, AL amyloidosis patients with 95% confidence intervals. In gray is the control patients, and in the pink is patients with ATTR with 95% confidence intervals. So the key beauty of this technique, I'm only sharing 3 results. There are reports from multiple other centers, an Australian study and a study from Japan, and studies also from the NAC showing very similar findings. Small number of patients, different radio tracers, different institutions, but consistently the same message. You see significant difference between AL compared to ATTR. Now let's look at the last slide where you see the TTR data in orange or pink there is closer to the controls, and that's also consistently reproduced from other studies. So Dr. Sorensen's study here looking at ATTR retention index, you can see that it's lower than AL but definitely higher than controls. This is our own data. So ATTR in purple, values are in between AL values, which are much higher, but definitely higher than controls. And we also have data with the new tracer, P5 plus 14. And in purple, you can see that the uptake is higher in AL, but also higher in ATTR. So are there inter-tracer differences? Something we need to understand. The consistent message is with the beta amyloid binding tracers, perhaps the signal in ATTR is low. That was our initial impression. But in this particular study that we're conducting, what I see in orange is the fluor-beta-pair, and you can see there's significant overlap. But now I'm thinking, look at the variation in the AL. Look at how wide those ranges are. And this is not just for us. It's for Dr. Sorensen's study, Dr. Emden's study, for our previous floor beta pair, and the current ongoing study. AL patients show variation, whereas the ATTR data are much tighter. So what does this mean? Is it a biology difference between AL compared to ATTR? Is it how the tracers perform? And these are some things that we really need to understand. Then this important study actually published by Sarah Cuddy from our group, from our ongoing longitudinal study, showed us several important points. So this was initial look at 45 subjects. We published this data. And what we looked at is patients with untreated AL, so-called active CA, patients who were treated for AL, the plasma cell dyscrasias treated patients are now in complete or very good partial response with the bone marrow in the green. And the third group is the patients who have systemic amyloidosis, but no evidence of cardiac involvement with normal wall thickness, normal cardiac biomarkers. And on the right side, same groups, you see ECV. On the left side is floor beta pair. So I just want to point out the following. Just think about this. So these are patients in whom the treatment is completed. They still have significant amount of amyloid in the heart. So we have not cured the patient. We have treated the bone marrow, but there's still a lot of amyloid there. And look at the ECVs, right? It's not a single modality. The ECV is showing very parallel changes, very high ECVs, more than 0.4 in this group. Now let's look at the systemic AL without cardiac involvement. These are patients where the biomarkers are normal, wall thickness are normal, conventional criteria, current day criteria say they have no cardiac involvement. Look at half of them in this very small study have floor beta pair retention as high as an untreated cardiac AL amyloid group. Again, something to think about. Why is it that some of these patients have that? Now, is it the tracer? Now, look at ECV. Same story. The normal cutoff is ECV is shown in that bottom green. You can see most of these patients have mild to moderate abnormalities in ECV as well. So this is telling us there's biologically something going on in these patients in the heart that we are not able to detect using echo or cardiac biomarkers, but maybe advanced imaging can give us some more information in these patients. Now here's just this image to confirm these two things. So both these patients, systemic AL, no cardiac involvement. And you can see one of them is PET positive and the other one is completely PET negative. So phenotypic heterogeneity, why these patients are resilient to manifesting cardiac disease, why disease doesn't progress is something we need to understand, why some of them have more severe heart failure, we just need to understand this. What about outcomes? We know that this data emerging, this is really a new field, unlike DPD, PYP, and HMDP. One of the initial studies showing that baseline floor beta pair uptake is predictive of outcomes. You can see low uptake, mild uptake, and high uptake in the bottom. We have very similar data from our ongoing study showing prognostic value with higher levels of floor beta pair compared to low or intermediate levels of floor beta pair. So to conclude, current available evidence and future directions, molecular amyloid binding tracers are really excellent diagnostic markers. I think they go one step beyond PYP, something we didn't discuss. With PYP, DPD, HMDP, the specificity, if you use grade 2, grade 3, is nearly 100%. Sensitivity is only 70, 72%. So there's still a group of patients who have the phenotype of infiltrative cardiomyopathy in whom we have not excluded amyloidosis. Those patients are currently going to biopsy for further evaluation. I think PET may put us in a one further step for diagnosis using non-invasive methods. You can look at this for all forms of amyloid. That's a huge advantage. You can look at systemic and cardiac amyloid burden, and I think we can stage this disease. We can stage this disease. Does the patient have one organ, multiple organs? How bad is this overall disease status? The challenges, most studies to date are case control studies. So we've taken patients who have amyloidosis, definitely diagnosed. Of course, the scan is going to be positive. Patients controls who have no amyloidosis, scan's got to be negative. We really need more studies in a diagnostic cohort. In a referral population, suspected amyloid, what does it show? PET uptake is prognostic, small studies, but we showed in the heart uptake is prognostic. Changes in response to therapy, we have emerging data, very similar to Dr. Fontana's data. It's unpublished, so I didn't share, but the SUV goes down at 6 months, goes down at 12 months. Is it a huge dip? No. But is there a change? Absolutely. So it's really very helpful for us to think that we are now looking at tools to see improvements at the myocardial level. So which Prentice criteria? Statistically significant drug-induced movement, nothing yet, maybe emerging data. Statistically significant improvement in clinical outcomes, we now have a number of drugs. Imaging biomarker is prognostic, small data, but I think we have two different groups suggesting that. And the last Prentice criteria is not met for this as well. So let me stop there and maybe bring on the panel for a discussion on cardiac PET. So before we go on to the questions, I just wanted to tell everyone Dr. Sarah Cuddy is monitoring the online questions. And if there's any online questions, Sarah, maybe you can interrupt. Okay, perfect. So I see Dr. Sorensen there and Dr. Emden both. So let me start with Dr. Sorensen and ask a question. Your group has done some of the initial work in C11 Pittsburgh B compound and also quantitation. So far I've only shared data with retention index and SUV, which are ratio metrics and which are semi-quantitative metrics. What do you think about absolute quantitation of amyloid tracer uptake using these tracers?
In many ways, I would really advocate that at least for these initial studies.
Hi, I got your message. I just wondered if you've got any dates. I sort of need to get back to
I'm sorry. I hear somebody talking.
Yeah,
on the
sorry,
telephone.
there's some background noise, so we'll get back to you in a second.
Okay, so the quantification, absolute quantification with PET requires dynamic imaging, and that's a step beyond what most sensors do. On the other hand, it is really powerful because that's the pharmacological basis of what the tracer, how the tracer binds and releases. Very powerful. The data so far that we have been looking at indicates that in the late stage disease where we have been looking primarily, there is not necessarily a huge difference compared to the semi-quantitative approaches that you showed before. On the other hand, what shows up with these quantitative approaches is that the uptake rate into the tissue of the small molecules, at least, I know that from carbon 11 PIB is directly proportional to blood flow, and that allows you to indicate when it is released over time, how much stays in the heart over time, but also the distribution volume of binding in the extracellular compartment. Turns out that the distribution volume of carbon 11 PIB is very closely related to the ECV. Which means that there is potentially additional information there that could be utilized diagnostically, but the real opportunity for these quantitative approaches is that would allow us to look at more details related to treatment evaluation. What happens when we treat the patients on a physiological level? But I would also really advocate that this is done with hybrid imaging, so that what is learned from these advanced PET adventures will be duplicated potentially into other and more easily available technologies over time.
Perfect. Yeah. Thank you so much. Thank you so much. So Dr. Emden, can you share with us some of your experience with floor beta Ben imaging at your institution? How are you using it? Is it research, clinical? What are your experiences?
Thank you so much. May I share a few slides, Sharmila?
We can hear you. Go ahead.
I'm not able.
Oh, sorry.
To...
sorry. No slides at this part. It's only just discussion.
Okay. So the main the core message of our ongoing work is that time matters in terms of dynamic acquisition. So the ability of discriminating light chain amyloidosis, not only the cardiac one, but also the extracardiac location by using delayed uptake evaluation, that is 50 to 60 minutes after the radiotracer injection, enables with 18-fluorine for beta Ben to achieve a potential non-invasive tool with a high accuracy to be now tested in a multi-center study which is ongoing in Europe. The evaluation up to date is that this radio tracer is the mirror of osteophile radio tracers in ATTR cardiomyopathy setting, that is able to looking at the delayed uptake to give us a specific and sensitive evaluation also in the early disease of light chain amyloidosis only.
Perfect. Thank you very much. Really excellent points. So let me now move on to Dr. Spencer Guthrie here. And you have a new tracer with I-124 labeled imaging. And can you share some points about that with us?
Yeah, I have to disclose I'm not a doctor. I just play one on TV. but thank you. Yeah, you wanted to know just kind of the status of that. So this tracer was, of course, developed with Dr. Wall, and we have a number of investigator studies going on, including with people in this room. We're talking about reproducibility a lot here. We're just completing a retest reproducibility study, and we've been in planning for a phase 3. So we're going through the regulatory agencies for that process, and that is in cardiac amyloidosis. And I would like to make the statement that I think thinking about these. We talk about cardiac amyloidosis a lot, but these are systemic diseases. So yes. AL and ATTR. Sorry. AL and ATTR. And I think we see, and you see from some of the images that, even in TTR, there can be a lot of organs involved. And, one of the challenges as you go through regulators is you have to have a truth standard. And in cardiac disease, there are truth standards that can be used, but in renal disease and liver disease, there are, the true standards aren't there. So these are going to be exploratory findings, but you have to, study it in phase 3 in cardiac amyloidosis.
Yeah, no, excellent points about the true standard, and that's we are always thinking about whenever I show the systemic disease and uptake in various organs, like what does that mean? Like do we have biopsy proof and other things?
Just one other point, because this was brought up before, is this idea of pre-symptomatic disease. So, of course, we've seen, and this relates to the true standard too. We've seen patients who have particularly cardiac uptake who had, normal PYP or normal MRI. So, we believe that this is, and some of those patients have gone on to get biopsy, this is a way to detect patients with really early disease and hopefully, treat them before they develop these symptoms.
Excellent, excellent. Thank you so much. So let me now move ahead to Dr. Andrew Stevens, and can you tell us something about your study, and then we'll end with a comment from Dr. Wall.
Yeah, no, thank you. I agree with a lot that you said. So we are embarking on a phase 3 study, a pivotal phase 3 study in the U.S. and Europe, looking at the point that you made, not patients who have known diagnosed disease, but people who have suspicion of disease, so that we can really look at how it changes. The regulators were very interested in how it changes therapy and how it changes the patient, the physician's decision-making process. That was very important, particularly in Europe. So that study will start enrolling very quickly. The other point that I wanted to make that we've heard a couple of times is the importance of dynamic imaging, is that this study will be doing dynamic imaging. We think that the differences that you showed with the beautiful work of Dr. Emden that separate ATTR from AL is a pharmacokinetic one, is that what happens is ATTR has a lower binding affinity and it falls off that fibril earlier. And so by really looking either with, and Professor Emden has published this in a wonderful way, is either looking with AI techniques or even more traditional modeling, you can pull out kinetic parameters, which really are quite dramatic in terms of separating AL from ATTR. So while our first phase 3 study will really be looking at kind of low-hanging fruit, which is AL versus everything else in a later scan time, where in these case control studies that you've seen is at 100% sensitivity and specificity, we hope to be able to also enroll ATTR patients and bring that back for an imaging study later, where we can really start separating out some of these kinetic issues. And so we're really very excited about this, and hope to go forward soon.
Thank you. I think I'll come back to your study design question during the next discussion. Dr. Wall, so you've developed this compound, which is now an excellent tracer.
Thank you, Sharmila. When we started off thinking about imaging amyloidosis, of course, we came from the AL world when TTR, as you mentioned earlier on, was not very well described or appreciated. And so we were very interested, and I think there's a real need to be able to understand the whole body amyloid biodistribution. This is not only true for AL patients, but some of the obviously much rarer forms. And I think it's becoming more apparent now that in ATTR patients, that the extra cardiac amyloid deposits may impact various things, sort of certainly quality of life, maybe response to therapy when we start looking at amyloid clearing therapeutics. Every site of amyloid deposition outside of the heart is going to serve as a competitive sink for the amyloid clearing therapeutic, and that will impact, I think, the kinetics by which the cardiac outcome metrics actually occur. So being able to understand how the heart's going to respond to a therapeutic in a trial needs an appreciation of the rest of the amyloid in the patient, right? And so I think a lot of trials suffer from this inability to appreciate changes, the kinetics of how different organs will change in response to these amyloid clearing reagents. And so you'll have patients who have very fast responses, but they get lost in the averaging because other patients don't. And that could be because all amyloid is being bound by these reagents. And so if you're focusing an outcome, a study just on the heart and not appreciating what else is going on in those patients, I think is a problem. So I agree with you completely that PET agents have the ability to be very sensitive with respect that they can probably with high accuracy detect pre-symptomatic deposits in all organs, particularly the heart. Also, you get the whole body information. The quantitative nature of PET-CT is remarkable and well established. And I also think now there are AIQ-based methods out there to get around this problem of subjective quantification based on ROI measurements, right? So I spend days and days, because I have no life, sitting in front of the telly actually drawing ROIs on every organ on 57 patients. But now there's people out there who have automated segmentation techniques. We've looked at them, we've evaluated them, we've compared me versus the AI, and the AI does really well. And so now there's tools out there as well to be able to segment the images and within 5 minutes generate all this quantitative data. And that's what you need in a trial in particular, is once you've got your baseline measurement, as Ahmed said, looking at that individual across time as opposed to cross-sectionally, you can get a lot of information with all these things put together. So you can be sensitive, quantitative, objective, and get the full picture of organ load. And I think all these imaging techniques, and I really do think that, to Rodney Falk's point, we're all looking at different components of the disease. And I really don't think we understand how all the things we're looking at and can look at tie together, both structurally and kinetically. So removing amyloid and being able to visualize that with PYP or florbetaben, and then waiting to see how the global strain changes, we would expect we would all sit in the room and go, oh, they should both change together by the same magnitude. But that's an assumption that we haven't had the ability to look at. So there's lots of complications, and I think the value is considering all these things in the right context and understanding, or at least appreciating the issues that each can bring and the problems we have with cross-referencing them all as well.
Yeah, excellent summary. I actually wanted to bring in Dr. Wechalekar and Dr. Angela Dispenzieri, and then probably Dr. Wechalekar as well.
So I just wanted to make one comment, and it is about the changes in response to therapy in AL amyloidosis. You mentioned about the retention index as well as ECV, that it doesn't really change at whatever time point, along with the hematologic response. But we do need to kind of look at it from the perspective of the pathophysiology, which is the light chain toxicity versus mass effect. And what we are talking about is all about mass effect, that what is deposited in the heart. But heart is not only about what is deposited, it is also about light chain toxicity, and that's why the cardiac biomarkers of NT-proBNP or whatever are more useful, or, at least, so far. So I think that was the only point I wanted to make.
That's really a great point, because we have no way of measuring this so-called cardiac toxicity. NT-proBNP and troponin T are not adequate measures, and Dr. Falk and others have taught me that you treat these patients, and as soon as the light chain level goes down, they start feeling better before you see any changes on echo or MRI or anything else. So clearly the patient is feeling better, looking better. We know something has improved, but we have no measure to quantify that, what the light chain toxicity is. I think microvascular dysfunction, maybe oxidative metabolism, so further studies of metabolic imaging may give us some more insights into that area. Dr. Dispenzieri, do you have any comments for us?
No, I think the only thing I would reiterate is, the time dependency that Vishali alluded to. It's so important because each patient's going to have their own kinetics. So it's not only light chain toxicity, but the light chains are so very different. And so again, looking at other markers to understand the toxicity and time, I think are going to be really essential.
Perfect. Thank you. So we'll thank you all for the active participation and providing some good insights onto this topic. So let's move on to the next session. This is connecting changes in amyloid through imaging with changes in cardiac events, functional measures, and health-related quality of life. So this session we'll first have Dr. Wechalekar go first, and then Dr. Sperry.
May I get the slide?
Thank you.
So thank you. That leads us very nicely into this session about quality of life, but it's a challenge because the data is sadly lacking here in this particular space. So in AL amyloidosis, in TTR amyloidosis, as we've been told, of the three critical components that we're looking for, feeling better, functioning better, and living longer, quality of life is one of the crucial things that we need to look at and look at more often. A lot of quality of life tools are out there to assess how patients feel. They've been used in a number of clinical trials. The common ones which we tend to use and have been used in AL amyloidosis are the SF-36, the URTC, and the KCCQ-12. As a part of the last forum, which was held here a few years ago, Dr. Sanchorwala led this program where we had a conceptual model of building quality of life tools for amyloidosis. And this is really important slide because AL patients have multiple different things going on at the same time. It's a rare disease, a long time to be diagnosed. It has a number of physical effects which we don't capture with our blood results, biomarkers, and imaging. Patients are extremely fatigued, and often that's their very first symptom. It affects their physical functioning quite substantially. And then we have got multiple different organs in different combinations for every single patient. So the same quality of life tool for a pure cardiac patient may have no role in a patient with bad GI amyloid or autonomic neuropathy. And trying to capture all of these is a huge challenge. We have moved some way because we've got a bit more data than we had at the last forum. I think there has been some more move in TTR amyloid than we have in AL amyloidosis. In all patients with amyloidosis, these are two studies. One is from Dr. Sanchorwala from the group at Boston, and the second is from our own study at the Amyloidosis Center. And you can see that when patients are diagnosed, compared to the benchmark, quality of life across every single measure that's assessed within the quality of life questionnaire is impaired. Pain probably is the one score where amyloid patients don't present, but everything else is markedly impaired when patients present to us at diagnosis. And certainly this is something that we don't track too well as we go through therapy. What happens when we treat these patients? Do they actually get better? Yes, we just heard now that some patients get into a CR and they feel better. The very first study came from Dr. Selden for patients who had an autologous stem cell transplant, and this is going back to 2004, I think nearly 20 years ago, where he showed that patients who have an autologous stem cell transplant actually feel better, but this feeling better takes quite a long time, as long as 12 months after transplantation when patients start to improve. And this is more recent data from patients treated with on the Andromeda clinical trial as published by Dr. Sanchorwala, either with Cybo-D or daratumumab Cybo-D, where you can see that initially the quality of life actually worsened in these patients. But then after the first 6 months, when they completed the chemotherapy, there appeared to be improvement in quality of life, which then seemed to sort of level off after about a year following treatment. So yes, there is some improvement in quality of life, but it takes time. We looked at this in our cohort of 900 patients who were serially treated with a bortezomib-based regimen over time to assess how does it change and does the response to therapy make a difference with quality of life. It's important to remember that the therapies that we are using for amyloid are not really targeting the deposits themselves. They're just simply targeting the precursor. And then it's kind of old-fashioned. We're hoping for the best, the protein to go away and for the patient to feel better. And this is what happened. Everything below the line is getting worse. Everything above the line is getting better across multiple different scores from physical functioning to general health. When patients were treated at 3 months, all the patients, irrespective of what their response was, felt worse. At 6 months, pretty similar, not a huge difference. But then at 12 months, what we noticed was that patients who had achieved a complete response to therapy and those who had achieved a near complete response or a very good partial response to therapy, they were the ones who started reporting feeling better across a number of different quality of life scores. But the patient who did not get a deep response, we did not see any improvement in their quality of life scores. So hematologic responses to therapy do translate into improvement in quality of life, but over a fairly long period of time. But patients who don't get this deep, complete response will really see no significant difference. And this seemed to translate very nicely into survival benefit. A 5-point improvement of the PCS was associated with a significant decrease in the risk of death. And for every single point in the trajectory of the PCS, the risk of death decreases. And you can see these different curves at the bottom, where we are looking at different follow-up at different time points, depending on the change in quality of life, as to how much improvement or worsening there may be in survival of a patient, depending on if the quality of life improves or worsens. So it clearly tracks to survival as well. But what about correlation of this with imaging? You know, this is just quality of life and outcome. The same group of patients that we reported with quality of life score also were included in our study looking at 2D strain that Rodney Falk presented at the start of this. We did see that there was an improvement in the longitudinal strain. This was in the same group of patients who achieved a CR. And patients who achieved a CR had improvement in strain, but also in everybody else, the strain either remained the same or worsened. It took time for the strain to improve. But patients where we saw improvement in strain and CR, there was a significant improvement in survival. So the same group of patients where we saw improvement in quality of life, we were able to show that there was an improvement in the cardiac function. And Marianna looked at this same cohort of patients with cardiac MRI scans. It wasn't in the 900 patients because MRIs weren't done in every single patient. But again, this is tracking amyloid load over 6 months, 1 year, and 2 years. And as you can see in the bar chart, a number of patients remained stable, but there was an improvement in number of patients where there was either stability or regression of amyloid, and patients with regression of amyloid did better. So where do we conclude? Baseline QOL is markedly impaired in patients with AL. We do see changes with the current therapies, even though they are not targeting amyloid deposits, but these improvements are slow. They're only seen where we have managed to eliminate the precursor, a complete hematologic response, and it still takes quite a long time. And it's this group of patients where anti-amyloid therapies, which can target fibrils, may make a substantial difference. Patients who show improvement in QOL are the same patients who show improvement in imaging by global longitudinal strain, by cardiac MRI scan, and these are also seen on a similar timescale. Improvements in quality of life, as well as imaging, are independent predictors of survival, but we need to look at this in a broader study. But there's one key factor which links all of this together. At the moment, we've got only one therapeutic measure, getting rid of the precursor, achieving a complete response to therapy. I think a combination of these endpoints and using those endpoints more often and studying them better is critical. Thank you. I think I'll hand over to Brett for the TTR side of things.
Thank you very much.
Thank you. Brett Sperry from St. Luke's in Kansas City. Thanks to ARC and Dr. Dorbala for asking me to speak on quality of life in ATTR amyloidosis. While there, the prior slides kind of showed an overview of quality of life in amyloidosis in general. Being from Kansas City, I think I'm contractually obligated to focus on KCCQ, so we'll get into that here. And while there's not a lot of data in AL amyloidosis with respect to connecting imaging markers and quality of life, there's probably even less data with respect to ATTR. But what do we know about quality of life and functional capacity? We know that patients with ATTR cardiac amyloidosis have impairments in heart failure, quality of life at the time of diagnosis with KCCQ scores in several studies, including ATTRACT in the 50s to 60s range, with higher numbers being better. 6-minute walk distance is also impaired. We know that as a marker of functional capacity. We know that functional capacity, low functional capacity at baseline does predict outcomes, negative outcomes. And we know that patients with hereditary ATTR, particularly those with V122I, do have worse baseline quality of life scores in 6-minute walk distances. This is interesting, looking at the different domains of KCCQ, and while all the domains are low, patients in this study have significant issues with physical limitation, social limitation, and symptom stability. And then we know that there's an association between treatment and improved quality of life and functional capacity. So do changes in CV imaging parameters correlate with quality of life and functional capacity? This is a figure from ATTRACT, which we all are very familiar with, looking at 6-minute walk distance and KCCQ overall summary score with improvements or better numbers versus placebo with tafamidis. And trying to link that to echo parameters, there's not a direct link there that I've seen, but there is in the supplement some information about echo parameters and some potential stabilization or improvement in echo parameters versus placebo there. So while not directly linked, these are potentially happening at the same time. What about gene silencer therapy? So again, a sub-study here Apollo A, looking at echo variables at 18 months in outcomes, and there's relative stabilization or improvement in echo variables in a subpopulation of cardiac patients here with siRNA therapy in LV wall thickness, mass, GLS, and diastolic volume and cardiac output, while there's also an improvement in some outcomes with respect to NT-proBNP and death or CV hospitalization, something that needs to be looked at in bigger trials. Also here, this is from the NAC in UK, 16 patients treated with siRNA therapy matched with 16 untreated controls, showing similar baseline cardiac MRI findings, but siRNA treatment was associated with stabilized ECV as well as 6-minute walk distance and NT-proBNP. So correlations here with siRNA therapy. What about antisense oligonucleotides? Similar type story. Treatment led to decrease in LV mass by cardiac MRI in a small study here in LV wall thickness, while there were stabilization and numerical increases in 6-minute walk distance and decreases in BNP. Again, these are things. Most of these are things that are kind of happening in parallel. Imaging findings are improving while there's some cardiac indices here that are improving. But what are some areas that we really need to look at? First, what imaging markers in ATTR cardiac amyloidosis do actually correlate with baseline quality of life and functional capacity? Second, what changes in imaging parameters like echocardiac MRI, nuclear scintigraphy, which we've spoken about earlier today, what changes there correlate with changes in quality of life and functional capacity? And then I think an important one is, are there differences in wild type versus variant ATTR with respect to these baseline factors and progression of disease, not only in hard outcomes, but also the quality of life and functional domains?
Yeah, some great points, especially there at the end, Brett. So it's a point that's been brought up in the Q&A a few times, that it's probably going to be a composite of biomarkers that might be the most useful. And that's already been shown in kind of the prognostic staging systems that we have. They've really been kind of focused around NT-proBNP troponin, with one from Matt Maher's group that integrates diuretic dosage and NYHA classification in ATTR. And that's what they're kind of scoring systems that we can use for prognosis. And do we see something like that integrating imaging biomarkers with these other functional biomarkers as a way to monitor response to therapy, monitor disease progression? And I think, Atish, some of your work has already touched on this, so I'd open up to you first.
I think it would be very interesting to integrate those with functional biomarkers. I mean, the one that we've done, I didn't present because there wasn't time, is 6-minute walk test in AL. And we can show that it improves with time. Again, we do see a trajectory of a decrease and an increase because the chemotherapy will impact the patient's outcomes. And we have also shown that patients who have a very poor 6-minute walk test have very poor outcomes. Patients who improve their 6-minute walk test by 33 meters or 44 meters, two different thresholds, will have significantly better survival. And that's the same as we've shown with NT-proBNP, where it's a response marker. So some markers may be easier to integrate than others. I think we do need to develop an amyloid-specific quality of life score where we're actually capturing something wider than what's captured in the heart failure questionnaires or just the SF-36 questionnaires, to understand how therapy impacts these patients and how these patients feel better over time. How one can integrate that with all of the biomarkers is going to be a slightly greater challenge.
I don't know if anyone else has any comments on integrating this or would like to add to that point.
So again, some few comments, that we did have this health-related quality of life working group from ARC, that has looked at it. Again, it was only for AL amyloidosis, has looked at all the instruments that have been published to really assess quality of life in AL amyloidosis. And the two instruments that have undergone strict content validation as well as psychometric validation in AL amyloidosis are PROMIS 29 and SF-36. So that is what is being proposed as possible PRO instruments to really incorporate in clinical trials for AL amyloidosis till we develop something more significant or specific. I think the other comment that I would like to make, which was, I think, missed in majority of these clinical trials, is the use of meaningful important differences in PROs or health-related quality of life. All the MIDs that we are proposing for KCCQ12 or SF36 or for PROMIS 29 are based on MIDs in other diseases and not specific to AL amyloidosis. So we do not know what the meaningful important differences would be for AL amyloidosis. Maybe 2-point difference might be dramatically important for patients with AL amyloidosis compared to ischemic cardiomyopathy. I do not know. In myeloma, the meaningful important difference is 10 points. But that may not be really, maybe in AL amyloidosis you require 15 points, maybe you require 20 points. But since it's a rare disease, these instruments have not really undergone MID validation or MID this. And I think that, again, composite endpoints and multi-domain endpoints are critical. And I know that ARC is working with, there is a working group also that is kind of trying to develop this, led by James Signoret. So.
Yeah, beautiful points. Thank you, Vishali. Dr. Murray.
Yeah, I think I learn the most often as a clinical researcher from our patients, and the one common question I think we all get every day is, hey, how am I doing? And I think we do a pretty terrible job at telling people how they're doing, or at least assessing all that. And I think, imaging can really engage patients in a very big way, is my sense. They really do like to see what's happening with their pictures over time. And this is a population, I think, of patients that is like the most engaged of anyone in the world. And we could, I think, do a better way to leverage, remote, PROs and assessments of patients over time, because at least our clinical experience, we're trying to raise our bar, at least at our site, like they're doing at the NAC, to really consistently measure what's going on with patients. It's a little disheartening also when you look at the data and realize that it takes 9 or 12 months for people to really start to feel better. It's kind of terrible.
Yeah, so I'm going to go to Dr. Masri, but first I'll go online to Dr. Angela Dispenseri and then Dr. Fontana.
Yeah, thanks. So really interesting conversation. I think, as we look at these questions on the board, we're right here. These are looking at comparing two surrogates to each other. And I guess I would make the point that we don't need surrogates correlating or not correlating. We need real endpoints that are going to be meaningful to, kind of get us regulatory approval and that are going to be robust enough so we can get drugs to our patients. So, if two things correlate, you basically say you don't need the other one, right? Because you have one. So one of the questions I would ask is, what do we need to do to take this to the next level?
Yeah, I think a great point, and probably we'll discuss in the next session. So Dr. Fontana?
Yeah, I just wanted to make a general comment about the correlation of imaging marker with functional capacity and quality of life, in the sense, in a way, I'm always surprised how, if you take, 5 patients with absolutely identical echo, absolutely identical CMR, you do all the possible mapping, but completely different from a functional capacity. But in a way, surprises me, but the other way, we're looking at so different things, in the sense that the functional capacity is the final common pathway of the interaction between very different systems, the skeletal muscle, the lungs, the heart, the neurohormonal activation, the autonomic dysfunction. And it also surprises me how little, in a way, we have investigated those mechanisms in amyloid, in the sense we use very kind of basic tests like the 6-minute walking test, which are great for clinical trials, are amazing, but really just tells you how far a patient can walk. But the functional impairment, the mechanism underlying functional impairment can be so variable because of the multi-organ involvement, and that I think we need to make a much more effort on first understanding the mechanisms and then understanding how the treatment do have an impact on the different mechanisms underlying the reduction in the functional capacity. But I'm almost like quite tempted to treat the imaging and the functional capacity like a completely different. We are looking at so different things. Trying to find correlation is very difficult.
Yeah, absolutely. I hope you're a grant reviewer at the next NIH study subsection meeting for my K23. I'll go to Dr. Masri. I know we're running out of time. One of the questions I do want to ask, if we have time, is anyone using cardiopulmonary exercise testing clinically, and are they integrating that kind of information? But first, I'll go to Matthias across hand up.
No, the quick point that I was going to make is that the placebo effect on PROs and quality of life is large. And so you can, with imaging, if you have the right imaging endpoint, you get actually largely get rid of the placebo effect. And so that helps you with your sample size with what you're doing. The second point is it's very hard to use PROs in a disease where you have a dip and then people feel better, because patients, their point of measurement is not when they were started the disease process or the treatment, it's where they felt the worst. And so you might see the effect on your treatment just because people are feeling better after they felt worse, not related to the baseline. And that's where the magnitude can be. So in TTR, it's much different, for example, because it's, in a way, a steady disease. You don't really dip acutely because of a treatment we're giving. So they're very two different processes for TTR and AL.
Beautiful points. Did I see a hand shoot up?
Oh, no, it's just a comment about, CPX. And in my experience, many of these patients have a number of musculoskeletal complaints. As we all know, in addition, or just from the fact that they're, at least in ATTR, they're elderly, or if they're quite debilitated, if they've got, severe AL. I will tell you that, and there aren't really many, I've done a lot of thinking about exercise in amyloid, and there's some echo studies and some CPX studies. I think there's at least 2 out there, and so we don't know that much. And I can tell you that I have, I did a CPX on an ex-NHL player with ATTR wild type, and I mean the results basically showed his symptom limitations were respiratory.
Yeah.
So I think it's unclear, and I think it's a tool. I just don't know how to implement it in patients this ill, but it certainly is interesting, and clearly the validity vastly supersedes that of a 6-minute walk, which is obviously more practical for a number of reasons.
Mm-hmm. So, Prem, and then I think we're ready to move on to the next.
Just a very quick point. Yet another way in which ATTR is so very different from AL, of course, is that when you treat ATTR, what you're doing is preventing further progression of disease, right? And that, from a patient's perspective, is really difficult to measure, right? You don't expect reversal of disease. Mm-hmm. You're preventing progression. It's very different from AL. Mm-hmm. So that's yet another reason that these measurement tools are difficult to implement in the TTR population.
Yeah, nice point.
Yeah, really excellent points. We may have to just move on to the next session, and we can take the questions in the next session as well. We want to get everyone out to lunch, so perfect. So the next session is really going to be mostly discussion. I just have 2 slides to set the stage for the discussion. So it's standardized imaging, standardization of imaging in AL and ATTR research, and how can we do it. And I think most of the points were discussed to some extent, but we'll come back to it. So the first question is something that we discussed earlier, standardization of diagnosis. I think a number of folks here participated in developing these diagnostic criteria. And on the right side is what we consider typical imaging features of cardiac amyloid, whether it's echo, MR, or with PET, it's more kind of early emerging data. Then how do we approach these patients clinically? And I think this is something that there are a number of algorithms, a number of different ways to do this. I just picked this one from Dr. Morer's publication. showing that if you have a patient with symptoms or any reason to suspect amyloidosis, the first step would be to look for a monoclonal protein. If that is all normal, because you have imaging features or clinical symptoms of infiltration, then you would proceed with bone avitraceous scintigraphy. And if that is positive, then you would see whether this is ATTR amyloid and proceed with genetic testing. If the bone avitraceous scintigraphy is negative, but your suspicion remains still high, this is something we brought up earlier, then those patients, you may selectively choose some of them to go ahead with endomyocardial biopsy. Now, on the other side of the screen, if your monoclonal protein evaluation is not negative, it's positive or equivocal, then these patients need an urgent referral to hematologists, particularly hematologists with expertise in amyloidosis, so that an early treatment can be initiated if it turns out to be something important. Then a lot of times there's going to be biopsy, whether it's a bone marrow biopsy or involved organ biopsy or a fat pad biopsy, for example. And then you get amyloid, you type it, then you make a decision based on that. So to simplify this, I just said there's 4 steps. So step 1 is to really suspect the disease, whether it's imaging, clinical symptoms, all of them. Step 2 is to look for AL amyloidosis. If that's negative, step 3 can be bone avitraceous scintigraphy, and in select patients, you do genetic testing in everyone, and in select patients, biopsy. If your AL evaluation is not negative, then urgent referral to hematology, maybe biopsy, and then you perform cardiac biomarkers with or without cardiac imaging. So I think it's a fairly simple approach. If you think about it, the algorithm may seem complicated. So I just want to stop there, go across the table and really understand perspectives from all of you. First step is diagnostic algorithm. How is everyone doing this in your own site? And then we'll stop and then go into what is needed for research from echo, SPECT, PET, and MRI. What do you need to standardize? How do you standardize these methods? I think the questions came up earlier. So maybe, Matt, could I start with you? How are you doing the diagnostic algorithm at your institution?
Yeah, I mean, I think everyone's doing quite similarly, and Claudio, to his credit, purported that, you could do all this in one day. You don't have to do it in stages. The biggest issue is, I'd say twofold. That is clinicians ordering a PYP without ordering light chains. We still see that at our institution. There are ways to try to avoid that, but I try to tell them that they'll get sued one day and I will testify against them. That usually changes their mind about doing that. And the other problem is, obviously, in the community, I'm not leveraging SPECT imaging, using planar imaging, and a lot of false positives with patients on, thankfully, just tafamidis, which costs them a lot but doesn't hurt them, but it's, still a problem. So those are the diagnostic things that I think are still problematic. It's education, education, education.
Perfect. Excellent. Anyone else?
To follow up on that, so we looked at our database of 2,500 patients with amyloidosis that were referred to the center. And of those patients, so we used to have about 5% false positive rates of referrals in the initial years, which was related to mainly to Congo red staining, that it was overstained slides and whatnot and whatnot. Now, in the last 5 years or 5 to 10 years maybe, 15 to 20% of patients are false positively referred, and most of them are related to PYP scanning. And this brings up the point of what Matt was saying, that, and Rick can comment to that as well, because he looks at all the PYP scans, that the SPECT versus planar, blood pooling versus 1 hour versus 3 hours, and it's a massive disaster in the community. Despite, what Dr. Masri was saying, that, you can just tell them over the phone, but when you have a referral base from all over the country, it makes it extremely hard. And do you just see them and rule it out? And that's what we do, because we don't want to miss a diagnosis. But
can I?
I would agree. I think Matt's already reported numbers, others as well, the high number of false positives from the community. The other point I wanted to make that's a point of, I think, confusion for cardiologists is interpretation of light chain testing. And not only do clinicians fail to order light chains, but they also are, well, as a cardiologist, I am not prepared to interpret a light chain assay through my medical training. And now we're trying to educate clinicians. And as many of us have reported the frequency of monoclonal gammopathy, or at least, kappa abnormal or elevated kappa above the, kind of the upper limit of normal is fairly common as people get older. And so I think that's another important problem. We're telling clinicians to order these tests, but we're not really providing them with sufficient guidance into how to interpret the tests. And we say refer to hematology, and we have great hematologists to refer, but many people in the country, don't have the access to such great hematology support.
Excellent. Dr. Soman?
Sure. I just wanted to make a quick comment. I think is an important change we made to our algorithm recently. That is, in a patient who has a fairly high clinical suspicion of amyloid, so step 2 is rule out AL, we do gene testing in everybody now. With this increasing recognition that there are some mutations that are not PYP avid, and having nearly missed, 2 P85 isoleucine mutations, we have now included gene testing in that complement of tests that are required to rule out AL amyloidosis. So it's not step 3 and 4, but step 3 is both.
Excellent point.
I just had a kind of a follow-up to what Rick and what Matt had said. So I think this is an education question too, and just wondering what the panel thinks about how you handle this. So I've seen an increasing number of referrals for patients who have a clear gammopathy, sometimes already have AL amyloids, and the PYP is positive. But then in the community, the assumption is that they have both, which is possible, as we all know, but they sort of proceed with tafamidis on top of the treatment for AL amyloid without confirming what's actually in the heart. So I think that's something that perhaps if you don't see a lot of these patients going through the algorithm, it doesn't necessarily, we don't really educate people on how they should be handling that. And just curious what the panel thinks would be the best way to approach that type of education for the community.
Thank you. Rodney, did you have a comment?
Thanks, Sharmila. I just want to perhaps turn this a little bit on its head. You've got suspect amyloidosis for heart failure. It's too late, right? So maybe obviously atrial fibrillation with an abnormal echo. But what I'm going to suggest is we don't really know currently in elderly patients with heart failure who have a negative PYP and an echo that doesn't look like amyloid, is there a contribution from amyloid deposition? Because we know in patients in 70 and 80, they've got a lot of amyloid in their heart at autopsy. And maybe it's just a partial contributor to heart failure along with hypertensive heart disease or coronary disease or so on. So I'm just asking the people here to think that maybe we'll be sitting here in 5 years' time and saying PYP doesn't pick up everybody because 20% of the contribution of this person's heart failure is due to amyloid. Most of it is something else, but if they didn't have the amyloid, they'd never have the heart failure. So I think we may be still too narrow. I know it's not the purview of this panel. I just want to raise it. Thanks.
Absolutely. I think that's a really great point. And at this stage, and we are scratching our head, all of us, I think many of us, looking at early disease detection. What is your reference standard? Endomyocardial biopsy is not the best method for early disease. You may or may not hit the amyloid deposit, and what you see on one sample of biopsy doesn't represent amyloid in the entire heart. So what is your gold standard to diagnose disease before there's overt clinical manifestations and overt structural changes? And I think that's a challenge, and I'm willing to hear from others. I don't know who went first, but maybe Ahmed.
Sorry, I just wanted to follow up. How do we define false positives in the sense of patients being sent to us? Most of my referrals come with abnormal PYP, figure out what's going on. I don't feel that these are false positives if they are, not truly amyloid, because I think the cardiologist or whoever is referring to me recognize that this is a mess and someone else needs to look at it. But the false positives that we have seen, which are like tafamidis-treated patients who don't have TTR, about 4 or 5, I would say, over the last 1 or 2 years, and we stopped tafamidis at that point in time. And so I think that's part of the story, correct? Like, we want people to recognize the disease more. There's going to be, of course, a downside to it, but how many patients are being actually falsely or like shouldn't be inappropriately treated with drugs versus how many patients are being recognized maybe they have a problem and then sent to other centers?
I mean, this is, I think, an expected problem we all know, we've tried to highlight, right, which is when you have a test that you're using in an amyloid center where half the patients walk in your door have amyloid, it looks really great, and then when you go to the community where, 2% of people have or 6% have amyloid, the false positive rates go through the roof, so you can't really fault everyone. Ahmed's point about that. I could envision, to Rodney's point and yours, a day where we're leveraging a PET isotope and, we're finding out that 25% of the patients who are 75 and above all are walking in with AFib, a little bit of heart failure, and or none of that, and have amyloid in their heart. We went from thinking it's a rare disease, and PYP made it's not so rare, and God knows what's going to happen when we start petting everybody,
I'm really glad. I was going to make that exact same point, and that Rodney brought up and you echoed. I was thinking when we were talking about PET, is it possibly too sensitive? Are we possibly going to be picking up all these amyloid deposits when we start screening 80-year-olds? So I don't know. I think certainly for following disease activity, but I think that's one thing. But including people in a trial, if they have PET uptake when they're 80 and they have minimal heart failure, who knows what that is?
So Rick, that is the next step. So first we prove pathophysiology and biology. If you can show that the ECV is expanded, there's a little bit of PET tracer. At this point, we believe that that's abnormal. The ECV should not be expanded with normal aging. So if we think this is not physiology, this is pathology, then what you do with it comes obviously next.
Right. Excellent point about integrating different imaging modalities.
Absolutely. I think single modality is going to be hard. So Spencer and then...
Can I just answer Grace's question because she asked the panel and I want to make sure. I'm so sorry to interrupt, but I wanted to answer. So I think that the issue of light chain interpretation, again, we can be educated by our colleagues in that we understand what normal reference ranges are with degrees of renal dysfunction, and I mean we're trying to kind of like widespread implement that in our assay interpretation. But I think there are going to be people who have coexistent disease. We already know the NAC has already educated us. Christopher was the first author on the paper that showed 10% of people have like almost grade 2 or 3 uptake, correct me if I'm misremembering, who have AL. So I think that, there's going to be that problem, but there's also going to be people who are going to have both diseases because, and they would have to be obviously biopsied. So we're trying to provide, through educational documents, clinical guidance, but I think we have a long way to go, as you suggest. Sorry.
Okay.
Yeah, I was just going to make a comment on the true standard for, pre-symptomatic patients. It's a huge challenge. So it's a challenge for PET agent where, we have a smaller sample, so there could be some bias, but there's, a lot of patients that have been imaged that have TTR polyneuropathy. So they have no cardiac symptoms, so they're considered polyneuropathy because the drugs are approved for cardiomyopathy versus polyneuropathy. And when they're imaged, almost all of them have cardiac uptake. And so some of them go on because they're confused because standard imaging doesn't pick it up, whether it's CMR or echo, and they will move on to get a cardiac biopsy just to confirm, but you can't do that in everyone, right? So how do you prove that it's amyloid? Because in the sense of a trial, it could be considered a false positive, even though it looks like amyloid, smells like amyloid.
Yeah, no, that's a great point, Justin.
And 2 points. I think there's like a step 3.5 for us. We get a lot of biopsy results that are Congo red positive, but they haven't been typed. And so that, really causes a tremendous slowdown because, you're allocating this, you're trying to figure out, can we get the block or do we have to biopsy them again, which is, which is certainly an issue. So then the second point, and about there isn't, we have some hypotheses about preclinical disease. I know, Dr. Quarta and others, and, in the Dallas Heart Study and from epidemiologic studies, we know from individuals that carry genes that predispose them to ATTR, specifically V122I offers an epidemiologic way or a more robust epidemiologic way, I should say, to try to understand these things. But the best we can say is that the hearts of individuals that carry a gene that might predispose them to ATTR, AL is, I'm not going to comment on AL, that the hearts do look different. I think that's the best we can say. But when you look at somebody in front of you, we're comparing a 9 millimeter septum to an 8.7 millimeter septum that reaches some statistical significance, and we can write a really elegant paper. But so I think that really, and it doesn't mean that those aren't useful. I think that's a key first step. But what we don't understand, or a key limitation to all of those studies, and guilty as charged, is that we don't know what, none of them were amyloid-based, right? We don't have ECV. Even if they had MRI, they're just, the morphological features. So I think that is a clear unmet need, is to understand, really what is going on in these hearts, and at best we can tell that there is a signal.
Yeah, perfect. Thank you so much. So I think we have 9 minutes. I just want to do 3 minutes per modality. Maybe ask Dr. Rodney Falk, what do you think is essential to standardized echocardiography for research applications?
Just precisely echo for research applications specifically.
How do we standardize it across sites?
How would I?
Standardization of echo for research.
I think in terms of standardization, I did skim over in my brief talk that when we're talking about strain, we were talking about a specific vendor that probably has the best, well used, but the best the technology for that. But I think that echo is a superb tool for research. I think that, as Cristina and many others have pointed out, we do need a central lab to look at this data. And like anything in medicine, if the test is not done well, the results are not going to be good. But nowadays, with the quality of echocardiographic equipment, with well-trained technicians, we can do good studies. We should be aware of the whatever percentage it is that should not be measured. Take that into account either with the exclusion criteria or in the central lab. And I think that doing that and being very careful about the technique, it's easily done, it's easily reproducible, as we've heard multiple times, and it's just a good tool.
Excellent, thank you. So let me ask Marianna if she's still online.
Could I just mention one thing that's afoot, and that is that I think imaging societies are interested in this. I know Roberto Lang, some people here are working with the American Society of Echo to try to create a repository leveraging AI as the core lab.
Yeah, we'll
but a lot of these images.
Yeah. Absolutely, great point. That's going to be one of the topics for the afternoon discussion. I don't know if Marianna is still on. Any comments about standardizing MR? If not, we will go to Michelle, I guess. Yeah, go ahead. Why don't you tell us what we need to do for standardizing MRI for research, what you're doing in your trial?
Yeah, so what we are doing for our trial is to have the same, we have chosen the site with the same protocol acquisition, scanner, contrast, and core lab. It's very important. And something that I was discussing also with Cristina before, it's having the cardiologists doing the MRI. I mean, this study is particularly successful because Marianna is the cardiologist, the PI, and also the person responsible to the MRI acquisition. So most of our patients are coming from her. But other sites that they rely on the radiology department that is not engaged at the same level of the cardiologist of the PI in the study, in that case, running a sub-study is much more difficult.
Excellent, thank you. So Dr. Prem Soman, any comments about standardizing PYP imaging?
So I would emphasize maybe 3 points. One is, make every effort to make sure that the uptake is in the myocardium, which means SPECT, SPECT-CT if you have, dual isotope imaging for people who perform that. But the principle is make sure it's in the myocardium. And then do AL labs conjointly with PYP. And I think unless the clinical presentation is overwhelmingly in favor of wild-type disease, I would say also do a gene test before saying that this is not amyloid.
Thank you very much. Dr. Sorensen, could you comment about what is needed for standardizing PET for research applications, amyloid PET for research applications?
I think that it would, since we are in a world where we have a variety of different traces and none of them settled as the gold standard as of yet, and none of them will at any time point be available at all hospitals. I think we could do very well in terms of trying to at least try to utilize one, at least two of these traces in a subset of patients. I also think that we would need to, for each individual tracer, that we will need to conduct these test-retest trials to understand how sensitive they would actually be in clinical trials. We can pick up even the slightest amount of amyloid in the heart, but does it matter? And how much is a useful change in the measurements that we are going to perform? All of them should also be investigated using rigid pharmacokinetic models so that we understand to the bottom what they do and how they work.
Absolutely, really great point, and we can probably close on that, which is test-retest. Very critical for echocardiography, for magnetic resonance imaging, SPECT as well as PET, because right now, as far as I know, people can correct me, the data that we have for test-retest for GLS or ECV or native T1 comes from non-amyloid patients. That's my understanding. And the question is, amyloidosis is a disease that impacts the interstitial space. That alters your T1 and ECV. Is the variability the same in a patient with amyloid? And this is very true for PET. And somebody brought up the question of protein binding. I showed you the variation of the PET traces with AL amyloid. Is the level of protein, whether it's circulating light chains or something else, somehow impacting the tracer uptake, and is that somehow adding to variability? We don't know. So I think repeat measurements, both repeatability and reproducibility of each of these techniques could be very, very critical before we start making decisions about response to therapy and trying to understand what is a significant change in any given metric. So with that, I think we are nearly at the end of our time. So I want to just thank all the speakers, all the moderators, and all the panelists for really a great discussion this morning. We do have a small break till about 1:05 p.m. We'll reconvene again for the afternoon session, where we're going to have really excellent further extension of this same conversation to how to tackle this disease and imaging markers. Okay, thank you very much. We'll see you all at 1:05.